Leave.
This
This is your
conference will
last
now be
icon. Clicking on
recorded.
the button, you will leave from the session. On the top right corner, you can find three icons: people, chat, and settings. People, which shows the attendees of the webinar, and the chat option, you can ask any queries over there. And in the settings, you can use this button to check for the settings for your speaker and microphone. So, as mentioned, kindly remain muted during the presentation so that the presenters don't get disturbed. And for any questions, you can ask me here, or you can write a message in the chat box. Finally, I would like to request you all to give a brief introduction about yourself before starting with your talk so that we can know about you virtually. And it would be great if you could let me know your research interest so that you can have a collaborations further. Now, let's move on to our today's session. The first presentation is by Dr. Adafi Ermans Ekop from Jebel City for Science and Technology, Egypt. The presentation title is The Synergistic Effect of Using Bacteriophages and Chitosan Nanoparticles Against Pathogenic Bacteria as a Novel Therapeutic Approach.
Thank you, Sylvia, for this brief introduction. I'll start sharing my
Okay,
screen right now.
okay. Yes, can you make it as a slideshow?
For sure.
It's okay.
Can you see it? Thank you. Okay. I am Arabi Irman Siaku from Dwell City, a graduate from Dwell City, batch 2023. I'm graduated um, from biomedical sciences major, medical sciences concentration. I've worked as a junior research assistant at the Center for Microbiology and Phage Therapy for 2 years. I've worked on various projects that help in getting rid of multi-drug resistant bacteria. We aim to find new methods, compounds, products that can help to be used as an alternative for antibiotics. I have two published research papers. The first is the synergistic effect of using bacteriophage and chitosan nanoparticles against pathogenic bacteria as a novel therapeutic approach, which we will go through today. And the second is about the lytic activity of bacteriophage against Salmonella enterica and its synergistic effect with the kanamycin. First, I'd like to go through a brief introduction about our subject today. The subject that I'm tackling today is a very important subject that is essential to our health. As we can see here, we can see that there are different bacterial infections that can be new to our ages, which is very risky to our public health. As we can see here, these infections can transfer among humans, animals, and plants. It can be on surfaces, it can transfer through our um, personal things that we use daily and transfer from one person to another. As we can see here, this person is having too much drugs that can have different types of antibiotics which he doesn't need that much. These different antibiotics can help in decreasing the person's immunity system and can damage our um, microflora and help in developing multi-drug resistant bacteria that can transfer among us humans and can transfer to plants and these poor animals. This issue has occurred due to the misuse of antibiotics, which lead to the development of multi-drug resistant bacteria. And let me tell you that this issue differs from one country to another. Some country might suffer from the presence of multi-drug resistant bacteria, inside their hospitals, which can develop due to the misuse of antibiotics, which can develop this multi-drug resistant bacteria that can transfer from one patient to another through the medical equipment that are used inside the hospital, which show us that it's very important that every medical equipment that is used should be sterilized before it's reused again. And other countries can suffer from this issue among people themselves. Some countries that are uh, poor in education, the people go and buy antibiotics from the pharmacy without needing them. This misuse of antibiotics can develop multi-drug resistant bacteria inside their homes and inside the public transportation system, which can lead to easily transmission for these infections among people. The issue in this multi-drug resistant bacteria that they have a high percentage of adaptation and they can spread among different types of people and living organisms inside our ecosystem. As we can see here, this human have used so many antibiotics that he didn't use, and through this misuse, multi-drug resistant bacteria develop and then transmitted to these animals. These animals can develop also multi-drug resistant bacteria due to the misuse of antibiotics in their food and their water, which can lead to the development of multi-drug resistant bacteria and their transmission to our food and to the sewage water, to all elements in the environment. As you can see here, this is a report that was published by the World Health Organization at the 3rd of November this um, month. It can show the statistical analysis for the different um, dangers that we are facing among our lives these days, such as the getting COVID-19 or having food safety hazards, antimicrobial resistance, and air pollution. This statistical analysis can show the rates of death among people and the loss in money, which will be massive. As you can see here, the rates of death by the antimicrobial resistance by 2050 will be 5 million human beings a year, and the loss will be about 100 trillion American dollar. So we have to be aware and we have to stop this from happening. The latest report was published yesterday, was the title of the antimicrobial resistance, AMR. We need to save millions of lives by having different alternatives for using antibiotics. They can say that overusing and misusing antibiotics put us all in the risk of antibacterial resistance. This can be result in producing harmful bacteria and damaging our beneficial microbes, unfortunately. As I mentioned before, it can spread from one person to another, and it can spread among people through poor hygiene and unclean facilities. We have to handle antimicrobial with care. We have to be very careful while dealing with these different kinds of bacterial infections. What are the previous solutions that are on the field right now? Some bacteria we can develop vaccines for them, but these vaccines cannot be used for a very long time because these bacteria keep mutating and they keep changing. So these vaccines are not effective for a long time. That's why we take vaccination every year and every few months according to the rate of the mutation for each bacteria. Bacteriophage therapy is a very good research field right now, but we have to be careful because once the bacteriophage is incubated with the bacteria for a long period of time, the bacteria start to form resistance for this bacteriophage, which can be very dangerous. About using this bacteriophage only for treatment. Some plants can be used, they have antibacterial, but they cannot kill bacteria with a very high concentration. They can kill a small bacterial infection, the bacterial infection in a very small dose, it can get rid of it. Probiotics is another field that's on research right now. They're using some natural products that have antibacterial activity, but it does not kill bacterial infections with a high concentration. So we have to find a new solution. Some new antibiotics are being produced nowadays by mixing different types of antibiotics. Some of them can be natural, like penicillin, and others can be artificial, but we have to be careful because these new antibiotics can be effective in their initial use, but after that, the bacteria can develop resistance for these new antibiotics, so they are not the solution that we need. Some research start to work on the probiotics and bacteriophages, but as I mentioned before, bacteria also can develop resistance for bacteriophages, and probiotics does not kill bacteria with a very high concentration. Some plants can also be used as oregano, cinnamon, and garlic. They have a high content of antibacterial, antifungal, and other useful substances, but they cannot be used as a treatment. They can be used for prevention against the bacterial infection and other infections that can be that we can be subjected for. There is there are some products that are naturally present around us that can be used, and we can use them inside our research with a very high concentrations without leading to site toxicity or damage, any kind of damage to our human cells, such as this is chitosan. Chitosan is a natural polymer that present in the crustaceans, as we can see here, the crab and the crab and other crustaceans. In some kinds of plants and fungus also, they can have an antibacterial and anti-cancer effect, but cannot be used alone. It's preferred to be used as nanoparticles, will have a higher concentration and more benefits from using only chitosan. Chitosan is natural, so it does not lead to cytotoxicity to the cells, and they are specific to the bacterial cells. They start to um, attack the bacterial cells. They start to change the polarity of the membrane, as we can see here, so they can damage the permeability, and the content of the cell can go to the outside, so the cell dies. From another perspective, it when it's metallic ion, I mean, when we use, mm, for example, silver nanoparticle in combination with chitosan, they can lead to the damage to the DNA of the bacterial cells and the production of the different reactive oxygen species, which can lead to eventually to killing the bacterial cells. This is about using the nanoparticles to kill the bacterial infections, but nanoparticles have a small disadvantage, which is it's very toxic to the cells, which means that they cannot be used for a high concentration or it will lead to a damage and a huge loss in the human cells. So we cannot use bacteriophages, we cannot use only new antibiotics, we cannot use other solutions alone. We need to have a new solution. Some new solutions are suggested that we can make a combination between different types of bacteriophages and nanoparticles, different types of bacteriophages and antibiotics, but we can face the same issue, which is the resistance to all these new solutions. So we need another new solution. What we can do now? The goal that we are tackling today in this paper that I'm presenting is to show that we have used different types of bacteriophages with a very small concentration to kill different bacterial infections and to keep the human cells safe. We have used also a combination between different nanoparticles and different bacteriophages, which can lead to the presence of synergy and the bacterial cells does not have enough time to develop resistance for these new compounds that we make. As you can see here, this is the paper that I'm going to go through today. We've combined bacteriophages. We have three different types of bacteriophages with three different nanoparticle chitosan compounds, and we recorded their effect against different pathogenic bacteria. The three compounds that we made are a combination. The first is a combination between chitosan and silver nanoparticle. The second combination is a combination between chitosan and bacteriophage. The third combination that we made is a combination between bacteriophage, I'm sorry, chitosan with propolis. What are the methodology that we go through in this paper? We had some quantitative and qualitative analysis for the data that we got, and we've made antibacterial tests for the compounds alone and for the compounds combined with the different types of bacteriophages. The results that we got are very impressive. We were very happy to see that the compounds that we made had initial indication that these compounds has changed from only chitosan mixed with a metal such as the silver that we used here into silver nanoparticles, which has a higher effective uh, effect than using them in their normal size. As we can see here above, we saw the change in the color for the three solutions, which is an initial indication for the formation of different compounds than the initial ones that we start with. As we can see in the below, we can see three different um, figures which show that the compounds that we made are in the nano size, not in their normal size, which is very enormous when compared. This is the quantitative data analysis that we made. First, we started to determine the wavelengths of each compound, and we found out that the three compounds have wavelength which is less than the 400, which is a good indication that the compounds that we made are produced successively. As you can see in the diagram in the middle, we have done Rx, which gives an indication for the percentage of each element inside the components. And as we can see here, this is an indication that the chitosan, which is a natural product, so we have a high percentage from the carbon, oxygen, and hydrogen. And this is a compound that has a silver nanoparticle, where we can see that the silver Ag um, has more than one peak with a very high concentration. So we know that this um, compound contain silver and a natural product. which confirms also that the compound that we aim to produce have already produced. The third diagram that we have here is an analysis for the size of the nano that we can see in the figure here. As we can see that the higher percentage of the cells inside the different compounds that we made ranges between 20 to 35 nanometers, which means that we have already succeeded to produce these new three compounds. Then we started to see their initial activity on different types of bacteria. On different types of pathogenic bacteria, we've used Staphylococcus aureus, we've used Pseudomonas aeruginosa, and we've used Salmonella typhimurium. As we can see here in A, B, and C, the disk diffusion that we made for the three different compounds, we can see that there is an effective zone. It's small, but still there is an effect. So we can indicate that there is an activity for these three compounds that we made. As we can see here, for the for the purple wells that we can see here, we've applied um, crystal violet dye to the cells that we've planted. As you can see here in A, this is the bacteria itself without adding any kind of treatment. The purple color that we can see here is the formation of a biofilm layer when the cells start to replicate too much and they didn't have enough nutrients, so they formed biofilm. In B here we can see a clear three cells, which are the bacteria with a very high concentration of the bacteriophage that is effective on it. As we can see here in C, this is the same three wells we can see here in B, but when they are left for a long time. A small percentage of these bacteria start to develop resistance for the phage, so they started to grow again, and they grow biofilm layer again. As you can see here in D, this is the thing that we discovered in this paper, where the combination between each of the three compounds that we produced with the effective type of the bacteriophage, they prevented the production of the biofilm layer. We've incubated this plate for about 3 days to be able to see the changes that occurred. The graph, the change between B and C occurred after about from 8 to 12 hours. Every different cell changed accordingly, while these indeed these three wells stayed clear for 3 days, and they didn't develop any turbidity or any indication for the presence of living cells or biofilm layers. This graph in E can show that representative data on the different um, compounds that we produce on different bacteria. This is quantitative data. As we can see here, we've had photos for the damage that occurred for the bacterial cells when they were incubated with the three compounds for about uh, 12 hours, yes, with half of the minimum elevator concentration. As you can see here, there are pores inside the bacterial wall, which can allow the organelles of the cell to get to the outside region, and then the cell will die due to these pores. As you can see here, there is a damage also, and these these debris is the intracellular components of the cell. So these bacterial cell has already died, and here we can see the same thing. So we can have an indication that if we produce different compounds that are a combination between natural products such as the chitosan, propolis, bacteriophage, and we have combined them with metallic nanoparticle, and we've added to them a small percentage of the uh, biofilm, from the bacteriophage. We can see that there is a synergy for a long period of time that can reach to killing all the percentage of the bacteria, and we do not have any leftovers that can develop resistance against these different components. This is the thing that we are showing today. We've shown, we can see from this research that the three compounds that we developed are effective on different gram-positive and gram-negative bacteria. This combination can be used and can be very useful in the future. I'm raising a campaign from here that we need to get rid of these multi-drug resistant bacteria by producing different other compounds that can have the same and higher and better effect on different types of bacteria. Thank you for your time. These are my references. Thank you. If anyone have any question, I'm open for it. Hello, Sylvia. Can you hear me?
Hello?
Can anybody hear me?
Yes, I hear you.
Okay, thank you.
Thank you very much, Dr. Agafi Erman for your presentation. Any questions? If you have any questions,
Hello?
please do ask here or you can mention in the chat box.
Okay. Hello, can I ask my question? Don't tell.
I hope there are no questions. Once again, thank you, Dr. Agafi, for your presentation. We are moving on to next presentation by Dr. Landry Bayela Biteta from MS&T Cameroon. The presentation title is Knowledge, Attitudes and Practices of Healthcare Personnel in the Management of Diarrhea Cases, Case of the Logon and Cherry Department for North Cameroon. For North Cameroon. Hello, Dr. Landry. Can you make it as a slideshow, please?
Good morning, everyone. You will
Good
see my
morning, Doctor.
presentation.
Yes, I can see your presentation, but can you make it as a slideshow?
It's good?
Yes, that's perfect. You can start now.
Okay. Thank you again to this amazing opportunity to give me to share about our experience and our job done here in Cameroon. Asante is in Cameroon national-based NGO that conduct many research and implement health implementations here in all Cameroonians. Then we want to share about knowledge, attitude, and practice of health personnel in management of diarrhea about a remote area of far north region in Cameroon. Uh, we just focus in this plane, introduction, methods, results, and discussions, conclusion, and share about recommendation of these presentations. Then, as we know, diarrhea disease is remained a rare public health problem. Then every year we have approximately 1.7 million of deaths occurring each year globally due to diarrhea. This is in particular in the sub-Saharan Africa. And then in Cameroon, diarrhea represents 30% of the reason of consultation in health facility. Is the third cause of death behind respiration infections and malaria disease. About management, WHO recommends first-line management of diarrhea in children under 5 by continuing feeding them, increase their fluid intake, and providing zinc supplementation for 10 to 40 days to prevent dehydratations. Therefore, here in Cameroon, Cameroon adopts the the IMC in 1999 with the aim to reduce the morbidity and mortality of children less than 5 years. However, diarrhea mortality and morbidity. However, pardon, diarrhea disease affects adult diarrhea and adult. Then, how care provided can be managing this disease in adults and a daily basis in health facilities. Several studies have been carried out on the community evaluating modern care knowledge, attitude, practice regarding diarrhea. Another evaluate mm, uh, knowledge, attitude, and practice of community health workers on managing diarrhea. There is few study in management in case of diarrhea by implementation of IMCI. Then we want to know this study was understand the find out the knowledge, aptitude, and practice of health personnel in management of diarrhea diarrhea in far north region of Cameroon. Then we conduct a cross-sectional descriptive study um, in the far north region. This study is carried out in far north region of Cameroon. Far north region of Cameroon is a very remote area of Cameroon near to um, Nigeria, Chad, and Niger. It is constituted in 4 health districts, namely Kuseri, Gulfe, Mada, and Makare. These health districts receive a large number of displaced people due to the Boko Haram phenomenon, terrorism, and constitute a favorite area for the development of diarrhea disease with recurrence of cholera epidemic and incidence of diarrhea in this area was in 31%. And then participant was all study. The study target population is the health personnel of the reception, hospitalization, and laboratory services of the health facilities. All health personnel on duty and present at the time of the interview visit but voluntarily agree to participate in the study were included. Study site. This was an exhaustive sampling of the all healthcare personnel in the reception, hospitalization, and laboratory service on duty and at the time of the interview visit. About source of data sources, a semi-structured questionnaire adopted from previous studies administered face-to-face on smartphone in open data kit format allow us to collect data on the WHO definition of diarrhea and its cause, diagnostic procedures, and management of diarrhea disease by trained interviews. Statistical methods. Data data collected on smartphone was directly checked and corrected before being sent to the server. Its data was analyzed on using CDC AP-Info version 7 and Microsoft Access 2007. The safety statistics were used to calculate the frequencies and percentage. The variable study were the social professional parameters of health workers and knowledge, attitude, and practice of per health workers in relation to acute diarrhea. About the result, a total of a total of 77 health personnel participate in the study with a 100% response rate that we show of whom 57% were a nurse assistants, 22% were nurse, and 5% were a physician, 30% other professional. It's important to know that the other health personnel was constituted by the laboratory technicians and community health workers. In our remote area, we have some health facilities who laboratory technicians are trained to consult and prescribe, and then for the community health workers. And then the most represented age group was the 20 to 40 years old, and sex ratio male to female was 3 to 2. And the most represented health facilities were the 6 categories. Then in our systems, we have 6 categories of health facilities. The sixth is uh, 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 um, the last part of pyramid. And then about knowledge of interviews people. When the professional were asked about definition of diarrhea, 78% respond that diarrhea is the passage of 3 or more loose or liquid stool in 24 hours. Within the others, 21% respond that diarrhea was characterized by an increasing number of daily bowel movement with large variability observed in the frequencies. Regarding the most common cause of diarrhea, the most common response were bacteria and parasitic infections. This table shows us attitude and practice regarding the management of diarrhea. Of the 77 health personnel interviews, 29% said they had a diarrhea management protocol, and 82 implemented it. We have here another 52% performs stool examination done 82% doesn't perform a stool examination before management a case of diarrhea. This is due to the lack of laboratory in the health facilities. So many health facilities in this region does have equipment for laboratory analysis. That is a very important uh, uh, factor to antimicrobial resistance. Another thing, uh, 82% prescribe antibiotic for all diarrheas. It's a very, very big problem. About antibiotic prescribed, we have micronidazole, amoxicillin, and cotrimoxazole were the most frequently prescribed in children less than 5 years, and ciprofloxacin and ceftriaxone in adult people. Then, to conclude our presentation, healthcare providers in Logan and Shari Department of Cameroon have different opinions regarding diarrhea disease, as well as on the management of its disease. To improve management of diarrhea, it would therefore be important to set up the standard document for management of heat diarrhea while ensure its implementation by the. Hello?
Hello.
Hello, do you hear me?
Yes.
Hello.
Yes, we
Yes?
can hear the
Yes, we
voice.
can hear.
Okay.
Yes.
Okay, thank you.
Thank you for your presentation, Dr. Landry Bayela. It was really an informative presentation from your side. Any questions?
No, ask question. Take your answer.
I hope there are no questions. We are moving on to the next presentation by Dr. Manuela Jadrovic from Croatian Veterinary Institute, Croatia. The presentation title is Regional Distribution and Diversity of Aspergillus and Penicillium Species on Croatian Traditional Meat Products.
Hello, everybody.
Hello.
Just a moment.
Okay.
I don't know how to share
Just
my
click on
pres.
the share button.
Okay,
Have you
screen
opened your
share.
slides? Yes.
Yes, but I can't see my presentation in that window.
Okay, first you need to open the slides, then click on the share button.
Just a moment, just a moment.
Mhm.
Here we are.
Yes, we can see the slides.
Okay, so I would like to greetings to all. My name is Manuela Zadrevec, and I'm working in Croatian Veterinary Institute in Zagreb, which is capital city of Croatia. And I would like to present you our work, regional distribution and diversity of Aspergillus and Penicillium species on Croatian traditional meat products. The intensity of fungal growth on the surface of traditional meat products and the variation of microbiota depend not only on the traditional but also on the controlled and variable production conditions, which are influenced by season because the temperature and relative humidity are not regulated and microbial filters on pressure barriers are lacking. Croatian traditional meat products include various types of dry fermented sausages. For
Hello,
example,
Dr.
yes,
Manuela. Sorry for the
yeah,
interruption. Can you please change the slides accordingly?
yes, I'm changing.
Okay, but
But
we're able to see on the first slide.
really? Sorry.
Yeah. Otherwise, you can
I
make it as a slideshow.
I will start again. It's
Okay, it's the first slide now.
and now is it
Now,
second?
it's the first
No.
one. Click on the second one. Yes, this is the second slide.
Okay. Okay. So I understand. Uh, so I would like to continue on the
Yes,
slide
please.
number 3. Croatian traditional meat products include various types of dry fermented sausages, and for example, kulen, paprika flavored salami and eastern sausages, and then dry cured meat products, which is prosciutto, pancetta, and ham. These products are produced without starter cultures on family farms or in households that differ not only in the recipes used, but also in the hygienic and environmental production conditions. The ripening phase of dry fermented sausages takes place mainly during the winter months. Prosciuttos and hams ripe longer, taking more than 18 months. These products not only ripen in the winter months but also during the warmer seasons, the average ripening temperature being mostly up to 20. Production conditions, example ripening temperature and time, relative humidity, and intrinsic product properties such as pH, water activity, and salt content affect the growth of desirable surface molds. However, these conditions also allow for the growth of undesirable competing molds. Molds on traditional meat products contribute to the specific smell, taste, and texture resulting the production of lipolytic and proteolytic enzymes during fermentation and the ripening, and retention of moisture of the surface. These surface molds also have antioxidant properties through catalase activity and the use of oxygen. They protect the surface from light and enable color stabilization, prevent acidity, and are involved in the formation of a crust composed of coagulated proteins. In addition, the surface molds protect against spontaneous colonization of unwanted molds, yeast, and bacteria, and prevent slime and stiffness of the surface. Production condition and intrinsic product properties affect the growth of desirable surface molds. However, these conditions also allow for the growth of undesirable competing molds. Undesirable molds negatively impact on the quality in terms of deviation of organoleptic properties and surface staining, and they can result in food safety issues, including mycotoxin contamination. Therefore, the aim of the present study is to first investigate into the fungal diversity of Croatian dry fermented sausages and dry cured meat products. Second, link this biodiversity data to the production technology, psychochemical parameters of the final products, and regional weather witnessed during ripening. And third, predict potential food safety issues arising from the presence of mycotoxigenic mold. That's how we designed our study. 250 traditional meat product samples consisting of 108 dry fermented sausages and 142 dry cured meat products were analyzed. The samples were produced in a traditional way on family farms and in households situated in 5 different regions of the Republic of Croatia, as follows: Central, North, Eastern, Western, and South region, as it showed in the map. The sampling was carried out during 2 years period to 2020-2021. During sampling, the data on ripening time were recorded in order to link them to the weather. The microbiota on product surface were analyzed after the determination of psychochemical properties of the sample in question. In all samples were determined water activity, pH, and salt content. Also, microbiota on surface was isolated and identified by classical and molecular methods. For classical methods, nutrition agars DG18, malt extract agar, and Chapek yeast agar were used and incubated at 24 degrees Celsius through 5 days. Molecular methods began with DNA isolation, following by amplification and sequencing beta-tubulin and K-modulin regions. Amplicons were sent to Microgen Europe. Traces were assembled using the segment program in the Lasergen package. A strain identification was performed using the basic local alignment search tool. Searches of the NCBI gene bank nucleotide database. In addition, the sequences were compared to the local sequence database hosted at the Westerdijk Fungal Biodiversity Institute in Netherlands that contains all available Penicillium and Aspergillus reference sequences. For genes detection which are involved in mycotoxin production, conventional PCR analyses were used and primers showed on the slide. We wanted to confirm genes responsible for aflatoxin, citrulline, and ochratoxin production. Genes responsible for cyclopionic acid production were detected by real-time PCR, and primers, probe, and temperature regime are showed at the slide. During the production of traditional meat products investigated in this study, weather factors such as air temperature and the amount of precipitation were recorded in all TMP-producing Croatian regions under study, using the data provided by Croatian Meteorological and Hydrological Service. And here are some interesting results. During this study, 20 different Penicillium and 17 Aspergillus species were isolated. Out of total number of isolates, 528, a significantly high number of Penicillium, which was 320, and 208 of Aspergillus species. This slide shows how it was distributed between traditional meat products. On dry fermented meat were isolated 127 Aspergillus species and 197 Penicillium species. At the same time, 81 Aspergillus species and 123 Penicillium species were isolated from sausage surface. The most frequent Penicillium species was as follows: Penicillium commune, Penicillium citrinum, Penicillium solitum, Penicillium nalgoviense, Penicillium corylophyllum, and Penicillium chrysogenum. Among Aspergillus species, it was Aspergillus proliferans, followed by Aspergillus pseudoglaucus. Which product contain more molds? Dry fermented meat surface contain average 2.3 molds and sausages 1.9 molds per sample. According to ecology and physiology of traditional meat product surface mold species, no statistical differences were observed in water activity, pH, and salt concentration between the two sampling years, as well as no statistical difference in smoke content and species diversity in traditional meat products. Nevertheless, the salt concentration in two analyzed meat products categories showed a significant difference. Dry cured meat products had a higher salt concentration comparing to than dry fermented sausages. Mold species identified in traditional meat product surface display based on the production region and weather witnessed during production shows that on both products in almost all regions Penicillium genus was predominantly, except in south region where Aspergillus genus is a bit higher than Penicillium genus. It can be explained that the climate in that region is Mediterranean with hot and dry summers, often compared to tropical and subtropical regions. Daily temperature often rises above 35 degrees, and also winters are mild and wet. That condition prefer Aspergillus species, contrary to Penicillium species, which prefer colder and more humid condition, which is characteristic of climate in other regions. 7 species Aspergillus Kreber, Aspergillus flavus, Aspergillus niger, Aspergillus westerdick, Penicillium citrinum, Penicillium commune, and Penicillium nordicum able to produce mycotoxins were detected. Penicillium commune, which is producer of cyclopyrazonic acid, and Penicillium citrinum, which produce citrinin, are the most common, followed by Penicillium nordicum, Aspergillus niger, Aspergillus westerdick, which produce ochratoxin, then Aspergillus flavus, which produce aflatoxin, and Aspergillus kreber, which produce stigmatocystin. The highest sum of mycotoxigenic species was found in the products coming from western region, uh, 24 dry fermented sausages and 14 dry cured meat, followed by those from the northern region, 12 dry fermented sausages and 70 dry cured meat, while the greatest diversity was seen in the products originating from the western and the eastern region isolated from dry fermented sausages. 5 different mycotoxigenic species. It was found out that approximately 50% of the isolated mycotoxigenic species possess all the tested biosynthetic genes essential for mycotoxin production. And finally, conclusions. In nearly all the studied regions, Penicillium species were predominant, except in the south where Aspergillus species were more prevalent. A significantly higher number of isolates was found on dry cured meat products as compared to dry fermented sausages, with a similar number of identified species. Environmental parameters of traditional meat product production, which affect the prevalence of surface molds, include the duration of the ripening process and regional environmental climatic conditions. Approximately 50% of the isolated mycotoxigenic species possess all tested biosynthetic genes essential for mycotoxin production. Regular monitoring of mold species and their mycotoxins on traditional meat products is of the utmost importance from the public health perspective, and the results of such monitoring can prove beneficial for tailoring production technology development. Thank you for your attention.
Thank you for your presentation, Dr. Manuela Chodravik. It was a great presentation. And any questions?
Thank you.
You can also ask the questions in chat box directly with the speaker. I hope there are no queries. Thank you once again, Dr. Manuela, for your presentation.
Thank you. Bye.
Bye. So the next presentation is by Dr. Shweta Parimita Bera from PP Savani University, India. The title of presentation is An Analysis of the Ecosystems of the Narmada River, Exploring Microbial Diversity via Metagenomics. Hello, Dr. Shweta. Okay, I can see the slides.
Okay. Can you hear me? Hello?
Yes, I can hear you, but we can see the web page.
Oh, okay. Wait just a minute. I'll share the. Wait, I'll just share the slide. Is it visible now?
Yes, it's visible and you can make it as a slideshow.
Okay, okay, fine. So, my greetings to everyone present here. And I'm Dr. Shweta Parimita Bera, and I'm a assistant professor at Department of Microbiology here at School of Sciences at PP Savani University, Surat, India. And the presentation that I'm going to make today is regarding the bioremediation of textile effluent. And this is actually, we have collected the isolates from Narmada River, which is a river nearby to the city from where I belong from. And we have studied the microbial diversity via metagenomics. I have isolated 4 strains, out of which one I have taken forward and done the optimization study, and also the complete degradation pathway. So yeah, and this is the objective of all the
Hello,
studies
Dr.
that.
Shweta. I'm sorry for the interruption. We are able to see your first slide only. Can you please change your slides accordingly to your presentation?
Okay, is it changed now, like I did the second one?
No, click on the second one so that we can see the second slide.
I did.
No, it's pointed on first slide only.
Oh, wait a minute, I'll do again then.
Yes, we can see the second slide now. So for every time you need to click on the slide. Okay,
Okay, okay. Hmm. I'll just put it
thank you.
in a slide share. Okay, fine, fine, it's fine. So as I was talking about, the objective of the study is to analyze the ecosystem of Narmada River via metagenomics. We also did the manual process in which we went on to do the isolation and screening of dye decolorization bacteria. So the is it visible now? Can the slides can be seen now? Okay. So the samples were collected from Narmada River, and this is the site from where we collect the sample. Clearly, the river is polluted with dye stuff, and the color is actually turning into red at times when the pollutants are very high in river content. Just a minute. Okay. So, what did we do in this whole thing was we isolated organisms from the river water, and those organisms were optimized and characterized to do the bioremediation process, and bioremediating specifically the textile dyes. So the very first thing we did was after collecting the samples was to analyze the physicochemical characteristics. For this, we did the basic environmental factors, which is pH, temperature, BOD, COD, TDS, TSS. We followed the APHA format for these things, and these are the analysis result. The color was quite reddish brown. It was pungent in smell. The pH was quite high. The temperature was higher. Every limit was crossing the given limits, which is standard set by the DOE. So this whole process, the beginning one, we did two things, that is treatability test. This was done using enrichment culture technique, and the second one was the toxicity test. This was done using uh, a series of parallel flasks with increasing concentration. The dye that we used over here was Prussian red H3B. That's one of the most commercially used dye for dyeing industries. So the very first thing we did was isolating the isolation of dye decolorization bacteria. So the simple thing that we follow is the serial dilution thing, and this was the culture was like we and medium which had about 100 milligram per liter of the dye, and the organisms were isolated to were inoculated to it and kept for about 24 hours to 72 hours. So this was the first sample that we did, varying dilutions, and then again we transferred it to the plates in which we also did with different other concentration of dye. This is the result in which we concluded that four of the strains were showing very good amount of color decolorization, and out of these like parameters we could conclude that there were four isolates that were of more importance because they were able to do decolorization to a greater extent, like more than 50%. Now the screening was done on the basis of this decolorization activity. Now the process for the same is followed by the normal criteria in which we do the decolorization percentage, that is the initial absorbance minus the final absorbance, and all of this was done at 545 nanometers. Okay, so this is the data for the four isolates that we included in our study. The final, like average, it was given, we took it for 48 hours, and average decolorization rate for the first strain was 96%. Second one, it was around almost 80%. The third one, it was 75, and the last one was 58. There were many isolates that we studied, but they were not giving a decolorization beyond 50% or more than 50% of the decolorization. So we excluded those things and like took the four main strains for further study. Yeah. After completion of these things, we did the 16s rRNA just to get the confirmation about the strain, and the result showed us that the strains belong to the family or the species from Pseudomonas stutzeri, Bacillus teclinis, Bacillus flexus, and Cocoria rosea. So these were the four strains that we then studied further. This is the phylogenetic tree after the 16s rRNA, and the darker one is indication of the four strains that we concluded or studied further. As the first strain, which is Pseudomonas stutzeri, gave 96% decolorization, we carried this strain for further study. Now this is the metagenomic result, the same thing when we studied about the ecosystem of the Narmada river and the species that were predominant in that environment. Out of that, the most complex or the most isolate were from Pseudomonas stutzeri. So basically this was abundant in nature and also were having very good amount of decolorization activity. Now this is the same thing from that. So once we were sure about the strain, we then again took the same strain for studying the optimization process. So we did it with again different varying dye concentration. Then we did it with all environmental and nutritional factors. So the first thing which we changed was effect of like we studied was the effect of environmental as well as nutritional conditions. The first was pH. As you can see from the slides, the best decolorization was seen at pH 8. In rest of the pH, like I took from pH 4 to pH 12, and out of it 8 was where the maximum decolorization activity was observed. Next it was the temperature, it was best at 32 degree. Similarly we did I did it for dye concentration, and 50 mg per liter was where the decolorization activity was maximum. Like I did it till from starting from 30 milligram per liter to 300, but the maximum was observed at 50. Now this aeration effect was something which was very new in the study, because usually what happens is microbes, or these degrading microbes, are basically very good at aerobic condition. But in our study we could, like I could conclude from our activities, that microaerophilic condition was optimum for this growth. So microaerophilic condition was in which the level of oxygen was reduced to like not the negligible amount, but a little lesser amount than the aerobic one, and that actually kind of facilitated better results. So yeah, and the reason behind it is the electron withdrawing nature of the azo dye and the resistance to the oxygen attack. So basically what happens is, like from the previous reports and like other studies, we could conclude that superoxides and hydrogen peroxides build up, like resulting in the byproduct. So they inhibit the oxygen, and that is how the oxidized form of flavins are formed, and that is why the oxygen toxicity affects the growth rate significantly. And that's why microaerophilic condition in our study was something which was new to us, and turning point where we can conclude that the reason why 96% decolorization can be observed with this Pseudomonas stutzeri. Next was the nutritional source. For it I took two things: carbon source and nitrogen source. In carbon source we did about uh, 8 carbon sources, sorry, 7 carbon sources, and in maltose the result was maximum. Similarly for nitrogen I took 5, and peptone gave the best result. Like in when nitrogen source was given with peptone, the growth rate was seen to be maximum. So the final conclusion: the best optimum condition for the growth was concluded at temperature 32 degree, pH 8, dye concentration 50 milligram per liter, aeration microaerophilic, carbon source is maltose, nitrogen source peptone, enrichment medium. Again, this is one something which we also changed. Normally we you go for mineral salts, but or agar, but then here we have changed it with phenethylene mineral medium. This medium actually facilitated a better result. So this was the final things how decolorization was measured, and this is the how the UV spectral visual results. This was the control, and the other one is the one after, like before treatment, like when we didn't use any dye, any organism to treat, and after the use of Pseudomonas stutzeri SPM1, which was our first strain, the result can be clearly observed. So the next step was to understand the enzyme profile of the whole thing. So basically three enzyme assay were studied for this: azoreductase activity, lactase activity, NADH DCIP reductase. These three enzymatic action can be clearly seen the increase in the percentage. Like azoreductase was 95% increased, lactase 76, and NADH 88%. So this was also an indication, like the organism was able to produce these enzymes for carrying out the degradation activity or the simple decolorization activity. So after this is the final like before untreated, the control sample, the parameters which I showed in the first one, and this is after treatment of 20 hours, and like using the culture, what was the result. Clearly we can see the reduction rate. The BOD percentage reduced to 85%, COD 90%, TDS 70, TSS 75, and TOC 65. So this was a significant reduction uh, uh, in the heavy metals or any like untreated sample. The next thing was to understand the quantitative analysis. For it I took three things: HPLC, FTIR, and GCMS. Now HPLC, this is the result for HPLC, which was before untreated sample and after treated sample, that clearly showed that there was a degradation. The graph can be easily be seen in this one. Then next was the FTIR. This was again similarly, it was showing the molecules in which the functional group can be seen, how at which and then specifically they are being discharged or eluted. This is the result for GCMS. The first one is the control one, which was before treatment, and the second one was the after bacterial treatment. So this GCMS, after GCMS, they like provided me with the name of the compounds which were produced after the degradation process. Like decolorization was the standard from where we could understand that okay, these are the organisms which show good degrading properties. But then degradation properties were checked by GCMS analysis, and the several peaks with this treatment showed us that the metabolites which were produced after it have significantly low toxicity as compared to the first time. So after this, this was one of the pathway that was proposed of how this Prussian red, which is around 774 molecular weight carrying, this could like undergo the pathway, and finally it will be like degraded out and degraded to better non-toxic substances. Now the final thing was phytotoxicity test. The tests were again, I have done this with plants. So the phytotoxicity test was done using two plants, that is Vigna radiata and Sycor erythrum. So I did it with three parameters. First was germination percentage, second one was plumule size, and the radical size. So again here we could easily see and differentiate that before treatment and after treatment. Like when the plants were shown in with water having like untreated one, there was no germination at all, because it already had high quantity of heavy metals and toxic substances inside it. And after the treatment, there was 80% germination in Vigna radiata and 90% in Sycor erythrum. So this was the result for the same. And the final conclusion of the whole this study was actually to understand that the bioremediation process can be carried out with the organisms which was predominantly present in the river itself, and they help in decolorizing as well as degrading the wastewater, specifically the dye stuffs. So the final conclusion was like, if bioremediation, like there are a lot of water treatment facilities available, the physical one, the chemical one, and including the biological one. But if you are aware of the organisms which have natural ability to do this, this can be utilized and can be done in urbanization for maximum expansion of water bodies and keeping them cleaner in for future consumption. So yeah, that's pretty much about everything, and this is the references. Thank you.
Thank you very much for your presentation, Dr. Shweta Parimita Bera. It was
Yeah,
a great
thank you.
presentation from your side.
Thank you so much.
Any questions?
For any queries, you can please write it me on the chat section. I'll be ready to answer those things. Okay, fine. Thank
Okay,
you so much,
so
Sylvia ma'am.
yeah, thank you. So I hope there are no questions. If you have any questions, please do not hesitate to ask in the chat box. And I'm moving on to the next presentation by Dr. Shafi Hassan from Jamhuriya University of Science and Technology, Somalia. The presentation title is Antimicrobial Susceptibility of Escherichia coli Isolated from Diabetic Patients in Mogadishu, Somalia. Hello, Dr. Shafia Hassan. Can you please share your screen? Hello, Dr. Chef here. Can you hear me? We can see that you are in unmute. Can you please speak out? Hello.
Hello, are you hear me?
Yes,
Hello,
I
are you hear
can
me?
hear your voice. Yes, we can hear your voice.
Yes. Hello, are you hear me? Hear your Yes, we can hear your voice. Yes. Hello, are you hear me? Okay, let me share my PowerPoint. Okay.
Yes, yes.
Yeah, organizer, please let me share my PowerPoint. Give me share icon. I can't share my PowerPoint. I don't know why.
You can share it now.
Okay, now it's okay. Okay, how screen? It's my screen. Hello,
Yes,
do you
that's
see my
perfect. You
screen?
can start. Yes, you can start now.
Hello,
Yes,
hello, do you hear
you
me?
can start now. Yes, we can hear your voice and your slides are visible. You can start your presentation now.
Okay, thank you. I will start. Hello, everyone. My name is Shafi Abdul Qadir from Somalia. I am from Jamhuriya University of Science and Technology. I'm the head of Medical Laboratory Science Department. I want to share with you my research entitled "And Microbial Stability of Escherichia coli Isolated from Diabetes Patients in Mogadishu, Somalia." Uh, as you first, my background: urinary tract infection are a major concern for diabetic patients due to the impact of diabetes on urinary tract and immune system. Uh, Escherichia coli is the most frequent organism that cause UTI in diabetic patient. Uh, this study I made to assess the microbial susceptibility of Escherichia coli strains isolated from diabetic patients in Mogadishu, Somalia. The main objective is to isolate Escherichia coli and make susceptibility to antibiotics commonly used as a treatment of urinary tract infection. The method is it was descriptive and cross-sectional study conducted in Hospital in Mogadishu, Somalia from November 21 to April 22. Clinkage midstream urine samples were collected from each patient participant that after we make consent and received consent to participate in the study. The samples were cultured on Kletaga on the samples were cultured on Kletaga. Then the samples were determined using biochemical tests to show that to check that Escherichia coli. There's other organisms that were isolated, but the most frequent organism was Escherichia coli. And then the samples were then subjected to AST or antimicrobial stability testing using Kalbai Bauer method or disk diffusion. And then that analysis was performed using SBSS version 20. This is the samples frame. First, we collected urine, and then
include, we observed to yellow color that shows urinary tract, that shows UTI as a lactose ferment, and then we use it as a confirmatory test or to biochemical test using API 20E. Then we we done AST using disk diffusion method, and we measure zone and produce it the minimum zone. Result is the overall balance of uropathogens among diabetic patients was 236 samples were observed, were isolated, Escherichia coli, these being the most common organism. And also we done logistic ration, asking the patients that the association between UTI and diabetic patients, and was significant associated to urinary tract infection. Then the result of antimicrobial stability were the most sensitive antibiotic is observed was colistin, 99.2%, and also followed by eminent, 99.2%. Also gentamicin observed 70% of sensitivity. The most resistant antibiotics was ciprofloxacin 100% and ofloxacin 90%, where ciprofloxacin observed 77.5%, amikacin resistant was 6%, while 58% was observed in ceftriaxone and 51% in cefepime. Therefore, the most effective organisms that we recommend to use in management of UTI was colistin, which observed 99.2%, and ibinum was 99.2%, and gentamicin was 7%. In the conclusion and recommendation, the study found high prevalence of uropathogens as observed 236 patients have a positive UTI were isolated to Escherichia coli. Therefore, also high resistant pathogens were observed. The most resistant organisms were ofloxacin, ciprofloxacin, amikacin, ceftriaxone, and cefepime. Colistin and ibinum gentamicin considered suitable empirical treatment for Escherichia coli as they are more sensitive organisms. And therefore, we recommend continuous antimicrobial stability monitoring, both community and hospital setting is recommended. But we like to mention our limitation was that the study was conducted in single area, that we don't represent, does not represent the whole Mogadishu or other hospitals, because we collected the samples in single hospital. For more inquiry, this study was published in Frontiers Microbiology. Everyone needs to more inquiry, please download. It's open access and you can download it for free. Thank you. Lastly, we need to thank the authorities of Jamhuri University and Science and Technology that give us the fund to conduct this study, and also we need to thank the authorities of hospital that allowed us to conduct this study. Because of them, we lack of them, we don't able to do that great achievement. Thank you. Any question? Hello. Hello, everyone. Any question, please?
Thank you for for your presentation. It's a very rich presentation, but I would like to know how do you calculate your site samples, please?
Repeat, please. I didn't understand your question.
Hello?
Hello, you hear me?
How do you calculate the study size?
Study sample.
How do you calculate the study size?
Okay, okay. Thank you. I understand. We use it a formula called Fisher formula, 1991, and we produce it to calculate the sample size, and we get the sample 384. But some sample is not responsible is we get it. The sample become 350. Thank you. We use it Fisher formula again. Fisher formula 1991.
Thank you very much.
Welcome, sir.
Thank you very much, Dr. Shafi Hassan, for your presentation. Any questions? I hope there are no questions. We are moving on to next presentation by Dr. Jaita Banerjee from University of Calcutta, India. The presentation title is Comparative Study on Nickel Biosorption Using Nickel Resistant Living and Non-Living Cerevisiae AJ208. Dr. Jaita Banerjee, you are the presenter now. Can you please share your screen?
Hello, I'm audible.
Yes, you are audible.
Okay, okay. Share my screen. Is it visible?
No, it's not visible.
Okay. Is it visible now?
Yes, it's visible. Can you make it as a slideshow?
Okay. I just enabling here. Is it completely visible as you instructed?
Yes, yes, it's
Okay.
visible, and you can start your talk.
Okay. 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 First of all, thank you for giving me this opportunity. Hello. Hello, I'm
Yes,
audible.
yes, you are audible.
A very good morning to the all dignitaries present here. Myself, Joyita Banerjee, Department of Chemical Engineering, University of Calcutta, India. My research topic is as entitled as Comparative Study on Nickel Biosorption using nickel-resistant living and non-living Cerevisia AJ208. But before coming to the core research part, let me tell you all what is nickel. why it is need to be removed, and why we have chose bioabsorption. Nickel is basically a transition state heavy metal. They chemically show variable valency and has a strong tendency of formation of coordination compound and naturally magnetorestrictive. Apart from this, nickel is also acting as a micronutrient in the chemical in the animal kingdom, and as act as an also a cofactor for the plant metabolic pathway. But the alarming thing is nickel is discharged from many industries at above permissible limit without being properly treated or totally being untreated. So it is alarming because it creates the potential imbalance into the ecosystem, which is potent dangerous to enter human life. So it is need to be removed. So these are some important properties of nickel, along with maximum contaminant level and the nickel permissible standard set by the international and the Indian prescribed limit in drinking water and the industrial effluent. And this slide is actually at a cluster of picture. The first picture is also a cluster of 6 pictures, which is showing the different type of industries which releases nickel basically above permissible limit. And the second picture is showing that the how nickel not only from the anthropogenic activity and from other various resources affecting the entire ecosystem and including human also, as respiratory distress, occupational distress, acute distress, chronic distress. So these are some nickel and it health hazards, nickel caused health hazards. Different type of exposure like inhalation. Inhalation exposure can be divided into the acute and the chronic exposure, and also the occupational exposure too. And other side of exposure is the thermal exposure and teratogenic exposure, which is happened during the pregnancy. It is very dangerous because it is passing to the offspring. And there's other health hazard I have mentioned here, that such as pulmonary edema, pneumocystis, nausea, nickel itch, cancer, and the chromosomal aberration and different level of exposure with the different level of exposure in the different set of time. So nickel need to be removed from the wastewater stream before discharging it into the freshwater bodies. Some available nickel removal method and their influencing factor are coming up. These are some remediation methods which can be divided into the physical method, chemical method, and the biological method. As we all know, these physical method and chemical method, or collectively we can say the physico-chemical method, which is a conventional method. It has some constraint because it is not very effective for a diluted solution. It is not very cost effective. It has huge operational cost. It can produce huge toxic sludge, but it is not even localized. So upon seeing this kind of a constraint, this kind of a limitation, bioabsorption seems to be a very feasible and a very cost effective and useful effective technique in a current days to remove different kind of pollutant in terms of inorganic and organic pollutant in our current days. So and these are some specific licking limits set by WHO and US EPA, and such as the also the Indian standard. So in India, Dr. Nagarajan et al. Reported regarding heavy discharge of nickel from electroplating industry in Madurai, India, up to 5569 mg per liter, which is very alarming. With consideration of these findings of the Dr. Nagarajan et al., we have decided or intended to develop the nickel-resistant microorganism with high nickel removal capacity at high nickel concentration from an aqueous solution, termed as a potential absorbent in the future days. So aim of our study is to basically carry out the experimental investigation to monitor and enhance the highest possible elimination of nickel from aqueous solution using our lab-made biosorbent as low-cost biosorption as low-cost biosorbent with testing of different parameter model such as the kinetic, isotherm, and the thermodynamic model for its potential applicability into the beta scale. So the materials and methods are representing here this kind of these materials reagent equipments, and this I have used all these during our experiment to for a run of our experimental study. For before coming to the code research part, let me tell you all that selection of the suitable microorganism is a far more important thing that we have previously mentioned that Dr. Nagarajan et al. With the consideration of the Nagarajan et al. Report, we have developed the 5000 mg per liter nickel resistant SABC AJ208. But initially, we have taken Bacillus rhamnosus MTCC 3161, which we have purchased from the Institute of Microbial Technology, Chandigarh, India, and the SLPC AJ208, which we have purchased from the Saket Pharmaceuticals, Saket UP, Uttar Pradesh, India. And we have actually studied MIC, minimum inhibitory concentration, survival tolerance study, and a bioabsorption capacity study to treat to treat and to screen which organism is better for to conduct our rest of the study. So basically, and that MIC study and by survival and tolerance study and the bioabsorption potential study reminded us that the Escherichia AJ20 is the best organism. So we have actually done the molecular characterizations with this strain, and we have deposited the nucleotide sequences of ITS1 and ITS2 of Escherichia AJ208 in the gene bank of NCBI, and the gene bank accession number of ITS1 and ITS2 is mentioned here. And after that, we have also prepared the dead Escherichia, because dead cell in bioremediation field is making a mark because of its nutrition independency and non-pathogenicity, because it is dead, it can't create anything harm. So we have also done this because dead cell, we have made the nickel resistant Escherichia AJ208 as dead by autoclaving it at the mentioned criteria. But we have also prepared the nickel stock solution, such as 6000 mg per liter nickel stock solution we have made, and it was diluted to get working standard solution. And with the mentioned specifically optimized media, we have maintained our bioabsorbent with this. And the bioabsorption experiment, how we have run that in a 100 ml conical flask with the 30 ml fermentation medium with 10 ml suspension at the 28 degrees centigrade for 48 hours. We have structured this bioabsorption experiment like this, and pH was adjusted with sorry 0.1 normal HCl and 0.1 in NaOH. Now, the experimental batch investigation of the adsorption are to be done, and the range of optimization parameter are mentioned here for living and non-living Escherichia AJ208. And we have also done the regeneration study of regeneration and the recycle study of the biosorbent to check the utility of this biosorbent in the industrial scale. And these are sorry, these are some analytical methods. With this kind of a formula, R means here the removal percentage of metal, and the Q is equal to the maximum adsorption capacity of nickel of the biosorbent. Using with this formula, these two formula, we have calculated all our necessary calculation, and we have also characterized the adsorbent, and such as molecular characterization we have done in terms of the phylogenetic analysis to screen and to screen the similarity and the dissimilarity of our mutated strain in with the parental Escherichia AJ208. Morphological characteristics other than phylogenetic analysis, we have done the scanning electron microscopy EDUC study FTIR and Raman study we have also done, and the zeta potential analysis we have also studied. And the same S. cerevisiae AJ208 was checked by ACM, scanning electron microscopic study, to evaluate the different kind of a surface morphology at the different experimental step. EDAX, also energy dispersive X-ray spectroscopy was also done to check the elemental composition in terms of nickel at the different successive stage. FTR and the Raman study are done or performed to identify the functional group the, and into responsible for the successful bioabsorption, and the zeta potential analysis also done to quantify the surface charge of the nickel listed Hysteresia, living and the non-living and the wild. It's a comparative study, and we have checked our residual nickel concentration with the atomic adsorption spectrophotometer, and we have also done the isotherm and the kinetic study and the thermodynamic study with the model using the pseudo first order second order model. Equilibrium study we have done with using the Langmuir and the Friendlich model, and the thermodynamic study also carried out. Now we have come to the result and discussion part. This process is schematically represent the which includes the making of nickel resistant Hysteresia AJ208 and the proposed nickel removal. Now this is the graph which represent the tolerance resistant versus concentration of nickel. From this graph, this figure, we can see that the nickel resistant Hysteresia AJ208 is survived up to 5000 mg per liter nickel concentration, as its MIC is 55500 mg per liter, which is also mentioned in the nearby in the chart. Is which is alongly placed in this same slide, and we can see that this, so I mean the figure represent the Hysteresia AJ208 is the best strain among the two strain, which is also that is Bacillus succulent MCC 3161 and Hysteresia AJ208. Among two strain, we have chosen the Hysteresia AJ208 because of this MIC value, and this chart is showing the results from which we have taken from the literature of the minimum inhibitory concentration for nickel for various microbial strain, with highlighted of our strain which we have used in our study. But one thing, I let me tell you all that we have chosen and taken Escherichia AJ208 for rest of our study as the potential bioabsorbent for not only basis of this MIC value. We have also done the bioabsorption potential study, and with the different successive level of resistance of these two strain, and we have seen that the only Escherichia AJ208 given the nickel removal percentage up to 60% before or the prior physical parameter optimization study. So we have chosen on the basis of the two ground of this strain for the rest of our study. Now the physical parameter study. So these two graph is representing here, that is the effect of concentration of nickel, initial concentration of nickel, and the time. Here we can see that the comparative figure of living cell and non-living cell, both graph is same, because in representing the comparative picture, as we can see that the optimum time after physical parameter optimization study for a better removal is 48 hours for living and non-living cell, but for the concentration of nickel, percentage removal obtained in terms of percentage removal, the non-living cell giving the higher percentage removal at the 500 mg per liter, as the percentage is 95, whereas the living cell is giving 90% removal, the same concentration. and the optimum pH of the living cell is 5, where the non-living cell optimum pH is 5.5. The temperature of the living cell is 28 degree, where it's for same for the non-living cell also, optimum temperature. And the volume of inoculum, optimum volume of inoculum of living cell is 12.5 ml, where it is 20 ml for non-living cell. And the percentage removal, ultimate percentage removal of nickel after physical parameter optimization study by living resistant nickel resistant Escherichia AJ208 is 70%, whereas the 54% nickel removal obtained from the non-living Escherichia AJ208. Now this is the scanning electron microscopic study. Whereas first picture where of Nicholastin Escherichia, we can see there is a prominent pore and the shrinkage, and the cell boundary become diffused and it get attached. So it is the Nicholastin Escherichia 208 after nickel resistant. Whereas the second picture is showing the cell become more attached, cell become more rounded and showing intact inner fullness. Whereas the third picture is showing which is non-living Escherichia AJ208 after heat treatment. It is the cell surface is more disintegrated, ruptured, and uncovered the intracellular space. So, and the fourth picture is obtained after the bioabsorption of the nickel-resistant Escherichia AJ208, because it percentage removal of non-living Escherichia AJ208 of nickel is lesser than the living cell, because the boundary is already diffused. So in the EDUX picture, whereas we can see EDUX picture with the elemental composition chart we have presented here, and from this first picture of nickel SLVC AJ208 showing the existence of nickel, whereas the second picture is showing is a uh, more prominent presence of nickel, but the third picture of the nickel resistant SLVCI is showing the nickel is present is nickel is present because is the cell is ruptured and the cell is disintegrated. So the nickel is present because it is a dead form of the nickel resistant SLVCI. And the fourth picture is showing that the nickel resistant SLVCI AJ208 in the non-living condition after bioabsorption, because the percentage of bioabsorption is less than the living. Here we can compare the after bioabsorptive phase of living Escherichia and the non-living Escherichia between 2 and 4 picture. From this EDUX picture and the chart is given here. And the zeta potential analysis. What is zeta potential? Zeta potential is a surface charge. The basically is we have actually studied in the wild Escherichia, nickel resistant Escherichia, and the non-living nickel resistant Escherichia. Here we can see the deviation of the spectral pattern. The zeta potential of wild Escherichia is minus 6.36 millivolt, whereas the nickel resistant Escherichia represent the zeta potential is minus 15.2, whereas nickel resistant non-living Escherichia AG20 represent the 8.63 millivolt zeta potential. That means the nickel resistant Escherichia zeta potential of the nickel resistant Escherichia representing representing it's a better biosorbent. So this is a FTIR study. Here the this three spectral pattern has been seen, and this spectral pattern and different peak has been seen with the and this chart is representing the spectral pattern with the different spectral pattern with the involved functional group of the FTIR study. And the Raman analysis, these are four pictures we have here given. The Raman analysis of nickel resistant Escherichia AJ208 before and after bioabsorption, and the non-living Escherichia before and after bioabsorption. Here we can see the prominent deviation of the spectral pattern. And these are responsible adsorption group, adsorption band, and the functional group involved in the Raman analysis. And literally we have come the isotherm model. So isotherm model we have studied to evaluate the equilibrium between the metal absorption and the adsorbent. And nickel resistant Escherichia, living Escherichia and non-living Escherichia both obeyed the Langmuir adsorption model. That means the adsorption was monolayer and happened in the homogeneous side of the adsorbent. Whereas the kinetic model has also been tested, and whereas the non-living Escherichia support the pseudo first order model means the adsorption is a physical adsorption, and the nickel resistant Escherichia living support the nickel pseudo second order kinetic model, which means it is chemically dependent on the chemi absorption. And we have came to the thermodynamic studies later. So that means for both living and non-living, minus or the negative G0 value means this is a spontaneous and the feasible adsorption, whereas the positive and the positive H and the positive S value, which means the entropy and the enthalpy, means it is random. It is defined the endothermic adsorption process, and specify the randomness, also specify the randomness of this adsorption towards the adsorbent. So this is a regeneration study. We have done this regeneration study to recycle and the reuse of this adsorbent to fit the adsorbent in the industrial scale or the better applicable structure. So living cell, and this is a comparative figure here we have used, and we have also used a 10 desorption agent, where we can see the HCl is the best desorption agent, and the highest percentage desorption obtained for 18 hours for living and non-living cell, and highest percentage desorption obtained for living cell. It is obtained from 0.15 mHCl, molar HCl for living cell, whereas ultimate percentage desorption obtained for non-living cell of HCl, and HCl ultimate concentration is the 0.1 mHCl is the ultimate desorption agent concentration. So we have also done the different use done the reusability graph for living Escherichia with 0.15 mHCl, and this signifies the adsorption capacity of living Escherichia AJ20 reduced to 69.33 at 10th uptake, whereas from the second figure it is seen that the dead Escherichia AJ208 or the non-living Escherichia AJ208 reduced up to 11% at the 7th uptake. So these are the optimized parameter and they are obtained magnitude for the living nickel resistant Escherichia AJ208, whereas the second chart is representing the same parameter value and their magnitude for non-living Escherichia AJ208, and their ultimate percentage removal of living cell is 70%, whereas the ultimate percentage removal of nickel is 54%. Why the differences between the percentage removal of living and the non-living cell represent? I'll state a short, brief narrate. I'll narrate bit briefly, because here we have seen that the. I'm coming to the previous slide. So here we can see from the living Escherichia AJ208, the prominent pore and the cell membrane getting diffused, and with the cell membrane getting diffused to suppress or to be tolerant into the huge metal stress, because the through pore diffusion is possible into the cell, and into the cell there is also the vacuole. which is a very much active for intracellular accumulation of metal in terms of nickel also. So the huge percentage removal from using living cell of Escherichia AJ208 is possible due to this feature of the living cell. Whereas many scientists supported that after rupture or the heat treatment of the nickel-resistant Escherichia, the when the cell is ruptured, it uncovers more intracellular space, more surface area, and the more functional group, which is the key point of uh, of removal of metal or the bioremediation potential of the dead cell, any of the dead cell, because these because these functional groups and the more surface area of the dead cell only facilitate the bioremediation of nickel in any point in other metal or here also. So, but the intracellular and but the other vacuole or the other accessory part is absent here. So, cell in a non-living cell is actually make it happen that it can bioremediate metal, but not like living cell. So that is the point of difference of living cell and the non-living cell removal percentage of metal. And we and the mainly the ion exchange and the electrostatic interactions is also responsible through the functional group that is residing outside the cell layer of any of the microbes is also the necessary is the mandatory part of the extracellular sequestration of metal of microbes. So we have came to the conclusion, and these are my concluding remark. And here we can we can say that the nickel-resistant Escherichia AJ208, both in living and non-living condition, is proved to be a potential bioabsorbent for nickel removal in at very high concentration, and from aqueous solution, which could be highly encourage the plan and design the strategic commercial exploitation as a potential candidate for nickel bioremediation. These are some references as the acknowledgement. Thank you.
Thank you very much, Dr. Jayita Banerjee, for your presentation. And any questions? If you have any questions, please do not hesitate to ask in the chat box. Okay, so I hope there are no questions. We are moving on to the next presentation by Dr. Augustina Nenna Okereka from Nigerian Institution for Oceanography and Marine Research, Nigeria. The presentation title is Evaluation of the Efficacy of Depuration System to Eliminate the Human Pathogen Vibrio vulnificus from Periwinkle tympanotonus fascicatus, harvested from Minorwig Creek in River State. Hello, Dr. Augustina. Are you ready to present your talk now? Okay. Hello, share face screen. Sorry for the inconvenience. The speaker is having some difficulty to log into the webinar. So give me some time. I'm going to communicate with the speaker and provide the guidelines to log in. So, and apologies for the inconvenience. Please stay connected. I'm very sorry for the inconvenience. The speaker is having some issues to join the webinar. So meanwhile, will go with the next presentation from Dr. Jane Ijeoma Ugochukwu from Enugu State University of Science and Technology, Nigeria. The presentation title is Formulation and Evaluation of the Antimalarial Activity of Artemether and Doxycycline Entrapped in Solid Lipid Nanoparticles. Hello, Dr. Jane. Hello, Dr. Jane. Is my voice audible to you? కొంచెం టైం ఇవ్వు. Hello, Dr. Jane. Do you have any issue to share your slides so that I can assist you?
Okay, I can.
Is it okay now? Hello, is it okay
Yes,
now?
I want to type in my. Yes, I'm talking.
Okay,
Yes, I
Yes,
can hear
yes,
you.
yes.
okay, then please share your slides.
Alpha bro. Don't go.
The slide is with you. Can you please show it? Can you please bring it out?
Hello, I request
Hello.
us to be on mute because we are able to hear multiple voices.
Okay. Can you hear me now?
Yes, we can hear two voices, that is Dr. Jane and Dr. Augustina. So I would like to know with whom I am speaking now. Is it Dr. Jane? Can you please confirm? Hello, Dr. Augustina. Can you please unmute? We are going to share your slides now. Dr. Augustina, can you please unmute and start your presentation? We have already shared your slides on the screen.
Can you hear me now? Okay. Hello.
Yes,
Yes, I
yes, we
am
can
Dr.
hear your voice.
Okay. Dr. Augustina.
Please
Name
start your
Odrende.
presentation.
Okay. I'm going to talk on evaluation of the efficacy of the depuration system to eliminate the human pathogen Vibrio funnificus from periwinkle Typonotus fuscatus, harvested from Minowe Creek in Rivers State, Nigeria. Vibrio funnificus is a human pathogen that is found in Austria and marine habitats. They are
Hello,
found in
Dr.
this region.
Augustina. Can you
Yes.
please let me know when to change the slides?
Okay, sorry. The first slide, that
Okay.
is the introduction. The introduction.
Oh, okay, okay.
Yes, I'm talking about the virus, that is the Vibrio funnificus, a human pathogen that is found in marine habitats. They are found in this lake where the temperature and the salinity of the area are low, thereby supporting their growth. Second slide. Periwinkle were introduced into a depuration tank. The depuration process involves placing trays of shellfish into a purpose-made tank, which is then filled with clean treated seawater using ozonized equipment. The depuration is one of the shellfish that is found in Rivers State, Nigeria. And the people, the community, they use it for their delicacy. The water was sent to the system through sterilizing equipment on a flow-through bag to allow sufficient aeration of water for the shellfish to function. The next slide. Materials and method. The sample, that is the periwinkle, used in this study were harvested from this Minowe Creek, River State, by October. It was collected in a sterile bank and transported to the laboratory in African Regional Aquaculture Center, River State, where they were prepared for analysis by first scrubbing and washing off the depth debris and surface contaminants using the tap water. Next slide. Next slide. Next slide. You can see the periwinkle sample in the decoration tank. This is a periwinkle. The next slide. The next slide. This is the decoration tank where the analysis was carried on. The next slide. Is also the sample of the periwinkle. The next slide. Can you go to materials and methods? Materials and methods. The materials and methods.
Thank you, Dr. Agustina, for your presentation. So, any questions?
If you look at the table, it shows the concentration of vibro depuration. At 0 hour depuration, the vibro bacteria was 4.6. At 96 hours of depuration, the microorganism was reduced. Next one. From these four, at 0 hour depuration, at 24 hours of depuration, then at 96 hours of depuration, it was reduced to 4.2.0 coliform count.
See?
Perform better in eliminating this contaminant as 96 hours of depuration. Unlike at 24 hours of depuration, the vibro count was high. At 48 hours, it was reduced, but at 96 hours, it was reduced drastically, which shows that depuration performed better remediation in these aquatic organisms. Thank you for listening. Thank you. Yes, question. Okay. Okay, can you hear me?
Okay, so we are moving on to the next presentation by Dr. Jane Ijeoma. Hello, Dr. Jane. Are you ready to present your talk now? Hello, Dr. Jane. Hello, Dr. Jane. Is my voice audible to you? Dr. Jane is having some audio issues. We are going to next presentation. So I request Dr. Nilay Solanki from Cherutar University of Science and Technology, India, to present the talk now. The presentation title is Clinical Decision Making in Infectious Diseases, Antibacterial Drug Utilization and Antibiogram Insights.
Hello.
Hello, Dr. Nilay.
Madam, just in 2-3 minutes, I'll start my presentation because
Okay,
still I'm traveling, so I'll start my laptop and then take.
okay,
Just
okay. So Dr. Nilay will start his presentation in 3 minutes as he's on the way. I request everyone to please stay connected.
Hello. Hello.
We are hello.
Hello. Am I audible? Hello.
Hello.
Ah, am I audible?
Yes, I can hear your voice, but I can see two logins from your side.
Just can you provide me the right address? Can you please?
You have to mute one login ID.
Oh, okay. So one is now it is
Okay.
muted.
Yes, I have made you the presenter.
Okay. So can you see my screen?
I can see the screen, but it's not the slides.
Is it now okay?
Yes. Please make it as a slideshow.
Now it is okay.
Yeah, that is okay. So I request everyone
Okay.
to be on mute because the it was a disturbance while presenting and talk from speaker side. So I request everyone to be on mute.
Okay. So myself, Dr. Neela Solanki, and first of all, I am very much thankful to the organizers for this conference. Congress, second Congress for providing me this opportunity. I am clinical decision making in infectious disease, clinical study. So this is important. As we all know that antibiotic therapy is one of the major achievement in modern medicine, and they are effective in different bacterial, viral, and fungal infection prevention as well as treatment. And here, individualized studies plays very important role, which is related with prescribing, dispensing, administering, and taking the medicine. And this intentional use of antibiotic can cause increased adverse reactions due to antibiotic resistance, which ultimately increase the cost of the treatment. So, and these antibiotics used without considering the administration of certain drugs and that's reduce its efficacy simultaneous along with the co-administered methods which generate related benefit. So the aim of this presentation uh, was this study was to identify the utilization pattern of antibiotics and the usage of the antibiotic two different study objectives. Exactly the drug that we have done during the study. So this is regarding the microbiology part. The study was conducted at our private hospital, Charles Specialty Hospital. Study period during 2019. The study design was observational process was and the study criteria were at least one antibiotic by prescribed in the inpatient department of the hospital or the hospital was segment CT was a study with a collection when demographic data were taken from and the basic case notes and the treatment charts. They were analyzed in this, using Microsoft Excel as well as some of the software supported softwares. So these are the results I am presenting. Social demographic data out of group patient we have collected, where 62 were patient were male and 94 were females. And this is the gender classification of the patient. And this is the age-wise distribution, where we have observed that out of 250 cases, most of the cases were in the age range from 20 to 60 years of age, from 0 to 60 years of the ages cases. And this is the condition-wise distribution by providing the different ICD codes for the different disease conditions, and along with that number of descriptions are provided for the different disease conditions. So this is the scene in the graphical representation. And here viral fever cases, lower respiratory tract infection, dengue cases were very high during the period of data collection. And for viral fever, it was observed that monotherapy and fixed combination was mostly where ceftriaxone was the most commonly utilized drug, and the combination was amoxicillin and glycolic acid for 21% of the combination. LTRI reports because of amoxicillin glycolic acid were highly prescribed well, as inflammation in ceftriaxone were prescribed in individual drug therapy. In dengue fever cases, we have observed that ceftriaxone was highly prescribed in 90% more cases out of 33 cases of dengue fever. But in COPD, where monotherapy, whereas ceftriaxone, azithromycin, and levofloxacin were highly prescribed by RTC, amoxicillin and glycolic acid were observed 48% of the cases. This is regarding the repeat recommendation for antibiotic, where only only 300 drugs antibiotics prescribed with the IV remaining were the oral. So in the oral utilization, the different types of antibiotic the biggest class of antibiotic, where cephalosporin and highly prescribed vaccines that usually ceftriaxone, then metronidazole and fomepizole are the most common prescribed antibiotics, which was also supported by the US study results. This is the overall utilization of the monotherapy for highest among all the therapies we was cephalosporin category of the drugs, and ceftriaxone was highly prescribed for 149 cases. Description were given of irrespective of the different conditions, followed by metronidazole, then azithromycin. And this is the different ITC code that for different ITC combination. And this is the same in the chart presentation. We also collected the data regarding the drug use other than antibiotics, like ondansetron was also prescribed along with over chewing therapy for antibiotics. Ondansetron 74% of cases, and bronchodilator deficiency. So these are the most widely and most common used drugs other than antibiotics that we have reported. This is the most important part of the study antidiagram, where we have observed that gram-negative bacteria, like Pseudomonas species, Acinetobacter, Klebsiella, and Citrobacteria sedi, where isolates where we have recorded, and now positive and ODS negative, so for species Candida albicans, as well as like we have reported during the study. The specimen that we have collected constituent positive tissue samples. In case of question, hello, hello. Is there any question for this presentation?
Thank you very much for your presentation, Dr. Nile. Any questions?
I hope I hope the presentation was fine. Is it audible?
So we are moving on to the next presentation by Dr. David Mead, Verizymes USA. The presentation title is Novel DNA Polymerases from Hyperthermophilic Biomass.
Hi, can you allow me to share the screen, please?
Sure, I'm making you the presenter now. You can share now.
Okay, hopefully you can see my screen okay, and you can hear me okay.
Yes, you're audible and the screen is very clear.
Okay, great. Thanks for this opportunity to present a story of metagenomic screening and of boiling hot spring viral DNA polymerases from hyperthermophilic biomes, and I should have included the word viral in there. So some of this information is over a decade old now, but it's a great little story. A partner and I had this idea to see if there might be some enzymes that could be of utility in boiling hot springs, mostly found in Yellowstone National Park with a permit. And I'm David Mead, the founder of Verizymes. We're a molecular biology reagent company developing isothermal amplification and other enzymes for molecular diagnostics. So almost everybody is familiar with DNA polymerases, but there is a pretty big difference between cellular DNA repair enzymes versus phage DNA polymerase replicases. And although this is not true all the time, it does have a trend of most of the phage enzymes have really unique properties that are better than for application purposes than cellular enzymes. For example, they tend to have strand displacement. Some of them are very high strand displacement enzymes compared to cellular enzymes. Significantly better fidelity, for example, with T4 polymerase. Phe29 is a really good example of a great strand displacement enzyme. Phe29 is another processive enzyme, and then analog incorporation is another interesting attribute. And all of these pictures are from Yellowstone National Park during our various trips out there to sample the boiling hot springs. This is a hot spring actually in Nevada. So as I've already mentioned, Phe29 has this incredible processivity capability going 70 kb and more. T7 polymerase is also very processive and it incorporates dideoxys. T4 is a very high proofreading activity enzyme, and MMLV is a very efficient reverse transcription enzyme. And the thinking was, the enzymes that were out there earlier, a little over a decade ago, uh, tended to be biased, did not have high fidelity or good affinity, had difficulty amplifying difficult templates, and so on and so forth. So we thought that it'd be interesting to see if we could find an enzyme in a boiling hot spring and improve it for biotechnology purposes. And we were going after phage enzymes and not cellular enzymes. And I don't know if you're aware of this or not, but viruses are the most abundant organism on the planet, and the estimate is 10 to the 31st viruses globally. And not all of them have polymerases, but the ones that do might be a nice new resource for their development. So this is Tom Schoenfeld. We are at a hot spring early in the morning by the Firehole River in Yellowstone with our sampling equipment. You can see there. And this is before next-gen sequencing had even been developed. And what we did was we concentrated the microbes in the phage, and then we separated them using small pore concentration devices. And then we had to make a library, and nobody really had done that before. So that was actually one of the harder rate limiting steps. We were able to make a very large library and cloned it in vectors. Again, this was before NGS. And then the Department of Energy's JGI did Sanger sequencing on that. And based on that data, we assembled and analyzed it and screened. We found several hundred potential hits for polymerases and screened the expression activity. And I'm not going to go through all that data. Some people will say, sure, how do you know they're really phage polymerases? If you do the right analysis, you can see, here's, for example, of a microbe that got through with this fluorescent staining on a membrane, and all these little green dots are phage. And then transmission electron microscopy also showed that we definitely have phage-like particles in our samples. And then when you look at the data, the sequence data that comes through from the two or three hot springs that we analyzed, there was a very high abundance of nucleic acid processing enzymes with a large number of putative polymerase hits. So based on that, we moved about 10 of those candidates forward and found one that worked really well. But it turns out after we were well into developing and trying to launch that enzyme as a product, next-gen sequencing came online and lots of data started coming out. And so we used our small chunk of the virus that we captured that had the virus, that had the DNA polymerase, to see if there's more of the virus there. And sure enough, we were found in the database an entire circular genome that is what we call octopus hot spring 3173 virus. We call it pyrovirus in one of our publications. And you can see it's got a clearly phage-like looking arrangement of structural proteins and then replication proteins and so on. So here we are sampling from octopus hot springs. This is where, and you can see it kind of looks like an octopus. This is Tom here watching his equipment, making sure none of the tubing breaks or anything. And based on that initial data, we did a lot of metagenomic mining for sequences and found some really interesting properties about our boiling hot spring virus that we stumbled on. And this is a little bit of that diversity and distribution story. It turns out there's a number of hot springs that had related viruses in Yellowstone, octopus conch, so on. And then the Great Boiling Spring in northern Nevada, and Little Hawk Creek is a little hot spring in California. So that just shows the geo-distribution. Some of these viral sequences aren't complete, but what you can see here is they all have the same kind of synteny and genome organization. Some genes are constant throughout, some come and some go. But in red here are the DNA polymerase genes, and you can see very large open reading frames for some of them, and 2 different reading frames for some of the others. And these are fairly large open reading frames that encode what looks like 3 proteins in one, whereas there's 2 here and 1 here. One of them looks to be a primase, one is an unknown function, and this little stub here is the polymerase. And that's the part that we went on to convert into a product, and I'll tell you more about that in a minute. So we actually found 4 intact viruses and published a really nice paper on them and their interrelatedness. And here you can see, in spite of their incredibly similar synteny and organization, the polymerases are wildly divergent in homology between the different hot springs. And we tried to work up all of them, but it turns out only a couple of them were really thermostable enough to do PCR. So we concentrated on those a lot, but these will come into play later on. You'll see why. So what is the host for this virus? Well, amazingly enough, the first year that we went and collected a sample and found our polymerase by sequencing, another group had done a metagenomic sequencing of the filamentous microbial community. You can see some of these pink filaments. These are Aquifocales thermocrinus, probably here in the outflow channel of this octopus hot spring. And sure enough, a huge number of hits, not to the entire virus, but a lot of hits to different parts of the virus were found in this metagenomic sequence sample. And then we did a really nice CRISPR spacer analysis using 4 isolated genomes, Hydrogenobacterium thermocrinus rubus and Jamesoni, and this sulfur hydrogenobium. And what you can see here are all these little triangles are CRISPR sites in the genome with homology sequences to different viral viruses. And so there's no doubt that Aquifocales and primarily thermocrinus are probably the main hosts for these viruses. One of the hosts that we compared the sequences to, thermocrinus ruber, which was collected from that exact same hot spring in 1998, had no viral CRISPRs in it. So a lot of interesting virology host interactions going on there. So it turns out there's an even more interesting story about the lateral gene transfer of this family A DNA polymerase, not only among thermophilic viruses, but also across the Aquifocales and across some eukaryotes, Plasmodium and Babsia. And I told you, the original first one we found was a large 1600 amino acid multi-enzyme protein. It turns out Plasmodium falciparum has another similar type of an architecture, and it's stunning. And these are closely related. We know from genetic analysis that they're related and belong to the same potential originating family. But it turns out this is a really unique exception in the literature. I don't think you'll find this anywhere else, that the polymerase from the complexia of Plasmodium falciparum is actually optimally active at 75 degrees centigrade. So that's probably one of the better examples of lateral gene transfer from a functional standpoint that you can find. So what about the applied enzymology? And as all of you are probably aware, polymerases are an engine of biotechnology for all kinds of applications. They've really revolutionized our ability to get at trace amounts of DNA and RNA and study it and manipulate it and detect it for all kinds of valuable applications. Well, during the development of this enzyme, we found that it was a really thermal stable enough enzyme to be a potent PCR enzyme, but to our surprise, it also had RT activity, and not just a little bit. It was pretty potent RT PCR enzyme. And so we did some work to try to develop it into a product, and it looks like it's as good as all the 2 enzyme systems that are out there, the Taq DNA polymerase plus a reverse transcriptase enzyme, PyroScript at the time. And it turns out this enzyme was also pretty good at amplifying tough GC-rich, difficult to amplify DNA compared to some of the other enzymes that are out there. Um, it turns out the fidelity is not nearly as good as fusion. It's better than TAC, but it's still not as good as fusion. Fusion is probably the best high fidelity enzyme for PCR on the market. So it obviously has some flaws that would be challenging to correct. Anyway, we also discovered that this enzyme was perfect for doing isothermal loop mediated amplification. Um, this is a factorial ability to amplify DNA in minutes, literally within 20 to 30 minutes. And this enzyme worked really well for that. For certain templates, it worked really well. It's got some issues, but if you want to know more about it, there's lots of publications showing how this works. It's totally unlike PCR. It uses 4 and almost always 6 primers that have these hairpin loops. And after it gets through the first round or so, you get factorial amplification. Literally, you can go from a couple copies to a trillion copies in about 15 minutes for most well-optimized primer sets. And so we developed that into a single enzyme. We called it an OmniAmp at the time, RNA and DNA polymerase. So it was really nice for isothermal lamp, room temperature. It would store really nicely for a couple of years as a lyophilized dried pellet. And the other nice thing about lamp is that most of the enzymes that are used for LAMP do not require extensive sample preparation. Literally just heat lysis with the right few chemicals in there, and away you go. So you can go from sample to answer in about 30 minutes with LAMP. So it's a really nice potential product for point of care detection of RNA and DNA viruses. And this just shows octopus hot spring again. That's the main hot spring, and then here's the outflow channel where we were collecting samples. And it turns out we sent a sample to a colleague who was working in the field trying to detect Ebola viruses in native mammals in East Africa. And it turns out it worked for, we had developed it obviously as a loop-mediated amplification technology that you would detect with lateral flow capabilities. And this was not the sample type that we got, but it turns out right when the sample came up, it was negative for the little rodent that they had tested. But this baboon decided to sit on a perch here, so got a really cool picture there. I'm sorry, it was West Africa, not East Africa. So it turns out this enzyme is fascinating as it is. It does not have any crystal structure, and it's very distantly related to any other polymerases. So we did a bunch of design of an experiment mutagenesis that I'm not going to tell you about. But my colleague, Tom Schoenfeld, who was the fellow who had this idea at the beginning, he went on to look at all those other polymerases that I told you about. And what he did was he took the best thermal stable one, 3173, and he chopped it up into the different motifs. And he used all of those variant polymerases and did a enzyme shuffling. He shuffled the various motifs in the various different ways and came up with an even better enzyme. And this just shows you some of the starting points. And he evolved the enzyme using its own parts from its own relatives to be an even better, faster RT-PCR enzyme. So that was a really nice piece of work. And then finally, to summarize, looking around in that same data set, we stumbled on another really unusual DNA polymerase that has a uracil DNA glycosylase native fusion built into it. And we're getting ready to publish that here, hopefully early next year. And it's a fascinating enzyme that removes uracils from DNA. And if you think about it, that makes sense for a high temperature organism where high temperatures are known to cause deamination of cytosine to uracil. So it would be a potentially catastrophic mutation. And this particular virus evidently is using it for some sort of repair property to prevent a lethal mutation from coming through. I don't want to show the data yet, don't want to hurt our publication, but it turns out UDG DNA Pol A's are found all over the place, but almost always in metagenomic samples in cold biomes, hot biomes, regular, temperature, uh, salt, you name it. They're all over the place. They've only been noticed but never described before in a couple of cultivated viruses. So this is a really fun story that'll come out soon. And the other nice, interesting thing is it looks like it roots at the very early part of the tree of Paul A. plumeraces. So it could be one of the ancestors that first arose, probably from a virus, obviously. So look forward to that story. And here's the authors on that if you want to keep an eye out for that. So I've had a lot of fantastic collaborators here over the year. Brian Hedlund, Marique Palmer, and Ryan Doss at University of Nevada, Las Vegas, and Eric Womack and Simon at JGI and University of Delaware. And all of this, the idea for all of this was Tom Schoenfeld's, and we had a lot of great people who helped make this happen at Lucigen, Scott, Yogesh, Began, Krishna, and Mike. And at Verizymes, Yogesh has joined me again, and Joy, Philip, Alyssa, and Kurt, and Aaron. And we've had some nice funding from the NIH and NSF. And we thank the National Park Service for allowing us to sample with a permit. Don't do it without a permit. It's not allowed. And be happy to take your questions. Hello. Thank you.
Thank you very much, Dr. David Med, for your presentation. It was a great presentation from your side. Any questions? I hope there are no questions. We are moving on to the next presentation by Dr. Jain. Dr. Jain, please do check your speaker settings because you are completely in mute and there is no settings updated till now. You have to check your speaker settings. I hope
Very interesting.
Dr. Jain is ready to present the talk now. So, Dr. Jain, can you please start presenting your talk? We can see the slides. Start from the first slide. Click on the first slide.
Yeah, I'm trying hard to start from the first slide.
Okay, just you need to click on the slide.
I've done that.
Yes, click over there on the first slide. You are on the fifth slide. So click on the first slide.
I think it should be network. I'm doing that.
No, there is no network issue. Just click on the first slide. You are on the fifth slide right now.
It's not moving.
Okay, then
The
make it as a
cursor is not
slideshow.
moving. Slideshow, slideshow. Stop sharing. No. Can you see? Can you see me now? Can you see my slide? Hello?
Hello?
Okay, that's cool. My name is Ugochukwu Jane, Dr. Mrs. Ugochukwu Jane Ijoma. I'm here to present on formulation, characterization, and evaluation of anti-malaria activity of activated doxycycline entrapped in solid lipid nanoparticles. Here are my outline. Can you hear me now?
Dr. Jane is having some speaker issues, so I request Dr. Laura Camila Carrera-Pez from Research Laboratory on Antibiotic Resistance Mechanisms, IMPAM UBA, CONICET, Argentina, to present the talk now. The presentation title is Studies of Mechanisms of Horizontal Genetic Transfer Associated with Antimicrobial Resistance in a Clinical Isolate.
Hi, can you hear me?
Yes, I'm just making you the presenter. Yeah, you
Okay,
can share the slides now.
okay. Can you see the
Okay.
poster?
Yes.
Okay. Okay. And now, can you see the poster?
Yes.
Okay, perfect. Uh, Good afternoon. Um, my talk is about the studies of mechanisms of horizontal genetic transfer associated with antimicrobial resistance in a clinical isolate. Like a general introduction, E. coli is the most common commensal bacteria in the gut microbiota. I I am hearing something. I don't know, like a echo. Can you hear me?
Can you hear me? I'm ready now.
Mm, hello,
Hello?
hello,
Yes.
Can I continue?
hello.
Hello?
Yes, we can hear your voice.
Okay, that's okay. Thank you. Can you see my slides?
Yes.
Okay. My name is Dr. Mrs. Jane Ijoma Ogochukwu from Pharmaceutical Microbiology and Biotechnology, Nigeria.
Hello. Hello, sorry for the interruption. Dr. Jane, the presentation should be delivered by Dr. Lara. Already she has shared her screen because you're having some audio issues. We have announced her talk now. So please wait for a moment till she
Okay.
finishes her poster presentation.
Okay, thank you.
Dr. Laura, please
Okay, I'm
continue with
here.
your poster.
Okay. So, the introduction was E. coli is the most common commensal bacteria that we found in the gut microbiota of both humans and animals. And this species has developed into a pathogen that is well adapted to its host. Um, One of these adaptations include the ability to acquire and disseminate antimicrobial gene resistance that could be transferred and acquired by a horizontal genetic transfer. And some of the mechanisms that we will know until now is transformation, conjugation, transduction, and the via extracellular vesicles. In these vesicles and by conjugation, these microorganisms could transfer plasmids that harbor the antimicrobial resistant genes. We choose an isolate of E. coli that was isolated in November on 2015 in Argentina and in Latin America from a patient with colon cancer, And in our study, we found that this strain belongs to sequenciotype 615, and this isolate has a plasmid that encode a gene of interest of MCR1, that is a gene that confers resistance to the last resort of antibiotic use in clinical, that is colistin. So our goal was to evaluate the adaptive capacity to extensive drug resistance to antibiotic through different AGT mechanisms in a sporadic clone of E. coli that we named M197036 sequenciotype 615. For that, we use different donor source. These strains we sequence in our laboratory by different technologies, and we analyze the plasmid of these strains, and we classify by incompatibility group, and we found some gene of interest that we also use like a markers of in our conjugation size, and we also analyze the conjugative system in these plasmids, and we also looking for the toxin antitoxin systems in the plasmids. Um, After the bioinformatic analysis of these plasmids, we start with the conjugation assays. For that, first we check the conjugation ability of this plasmid in our laboratory control strain, that is a G53 E. coli. And then the same experiments we did in our strain in a study, the M197036 E. coli, and in our strain also we did some subsequential conjugations after the first conjugation, and we did some experiments with transformation with a small clone that we did in our laboratory. In this case, the clone harbors a small plasmid that has the gene of AADV that confers resistance to gentamicin. To all our transformants and transconjugations, we characterize this by genotypic detection by PCR. Then we did some antibiotic susceptibility test, minimum inhibitory concentration test, and then these transformant and transconjugations were tested by maintenance assays. We did these assays without antibiotic pressure at the first and 10th day, and we checked all these by PCR. And finally, we did some characterization and EV extraction of the native strain and one of the evolved strain that I will explain there. The AVs were characterized by three techniques. The first was a global quantification of proteins using a kit, the microBCA. The second one was a dynamic light scattering, and finally by transmission electron microscopy. Then the content of the vesicles were analyzed in base of two characteristics. The first one was the protein, and the second one was the DNA content. Um, First, the results shows that the laboratory control E. coli G53 was able to acquire all the plasmids. So we checked that these plasmids are we can conjugate this plasmid in vivo, and the transformation assays then shows that our strain were able to acquire the small plasmid that harbors the AADB cassette gene, and our strain were able to acquire all the four plasmid that we tested that harbor different gene of interest, and we found that has a higher conjugation efficiency compared with the laboratory model. Um, and the subsequential conjugation that we did, we obtained two more transconjugants. The first one that we named Evolve one is the transconjugant that was able to acquire the two plasmid in the same time from the clinical isolates, and also harbor the native plasmid of the strain. And the Evolve 2 was the strain that was able to harbor the three plasmid, the native, the two that acquired by conjugation, and the small plasmid that acquired by transformation. And by PCR, we check that all our transconjugant and transformant, we detect the gene of interest in each of these strains. After that, we check by antibiotic susceptibility in antibiogram and minimum inhibitory concentration, that this strain was able to become extensive drug resistance, that is almost resistant to all the antibiotics that we tested. And then our maintenance assays showed that this strain at the first days was able to maintain all the plasmid that acquired, but at the first day, at the 10 day, these strains lose a plasmid that harbors the black KPC2 gene, and the others plasmid were able to keep it, but the plasmid that acquired by transformation at the 10 day is lost for complete. And in this graphic you can see the maintenance assay for the two strains that we name Evolve. The first and third bar show the results for the Evolve strain. One that is that harbor the two plasmid that acquire but conjugation aside, and the blue shows the maintain of the plasmid that is native in our strain. In this case, at the first day, we show that this strain was able to maintain almost in a 100% the plasmid that is native of the strain, and the plasmid that harbor the BLA ending M1 gene, and lose in a 10% the plasmid that harbors BLA CTXM15 gene. And at the 10 day, the pattern of maintenance of the native plasmid, the other plasmid that harbors the BLA ending one gene was maintained, but the other plasmid, this strain lost almost in a 60%. And in the second evolved strain that is represented by the second and fourth bar, we can see that the pattern changed absolutely, because at the first day the plasmid that harbors the gene BLASTXM15 was lost for almost for complete, and the small plasmid that the strain acquired by transformation was lost in a 10%. And at the 10th day, those both plasmid lost for complete. And the strain just maintained the native plasmid and the plasmid with the gene BLA in the M1. After that, in this part, we can see the results of the extraction and characterization of the EVs from the native E. coli. In this case, we found by dynamic light scattering that these EVs has an average size of 154 nanometers and a polydispersion index of 1.194. In the TEM results shown EVs with double membrane, spherical shape, and different sizes, and by genotypic detection, we found that the DNA that we obtained from these EVs, we found the MCR1 gene in this DNA, and then by proteomic analysis, we found that in our EVs, the localization of proteins was distributed like the major part of these proteins are from the membrane, like outer and inner, and 30% in the cytoplasm and 7% in periplans. Then the analysis of biological process was associated. The major of the proteins were associated to transport and metabolism on carbohydrates, lipids, proteins, and nucleic acids. And also we found, that was very interesting for us, that in these proteins are the MCR1 protein, that is the protein associated with the resistance to colistin. And then we did the characterization of EVs from Evolve 2, that is the strain that harbors inside the native plasmid of the strain, but also the plasmid that acquired by conjugation that harbors the BLA-CTX-15 gene, and the plasmid that harbors the gene BLA-ENDM1, and that harbors the small plasmid that acquired by transformation, that harbors the gene AADV gene that gives resistance to gentamicin, that is an aminoglycoside. In this case, we found by DLS that the average size of these EVs was the 174 nanometers, and the polydispersion index was of the 0.2. And by TEM, we found that EVs has the characteristic shapes with double membrane, and we found something special in these EVs that I don't know if you can see, but the morphology is very characteristic, because we found the like, you can see like a big EV, and inside are small EVs, and this is related with the biogenesis of the EVs. And then we found in the DNA that we obtained from these EVs that there was positive for BLASTX in 15 gene, AADV gene, and MCR1 gene. So we conclude that the results show the genetic plasticity of a sporadic E. coli clone, such as ST1615, demonstrating that this type of isolates could play a very important transitional link in the clinical dynamics and evolution to multi-drug stream drug resistant phenotypes within the nosocomial niche by acting as reservoir of gene-resistant antimicrobial that could then be disseminated by different mechanisms on HGT. Thank you so much. Any question?
Thank you very much, Dr. Laura, for your poster presentation. Any questions? I hope there are no questions. We are moving on to next presentation by Dr. Jain. Dr. Jain, are you ready to present your talk now?
Yes, I'm ready.
Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm ready. Yes, I'm
Okay, please share your screen and start your presentation.
Can you see my slides?
Can you see my slide? Can you see my slide?
No?
Please, you can share
Please,
it
you
over
can
there.
share it over there.
It's still loading.
There was a resound from your side.
Can you see it now?
Can you see it now? Can you see it now? Can you see it now?
No?
Please, can you share
Please,
it
can
from
you
your
share
side?
it from your side? From your side?
Okay, please send your slides at our email or WhatsApp.
I've done that.
I've
Okay.
done that.
Okay, then we'll share the slides and just you have to explain. And let me know when to change the slides. And I request that there is some issue from your side that we are able to hear twice your voice. There was a resound.
Can you still hear me?
Can you still hear me? Can you still hear me?
Yes, I can hear the voice, but I'm able to hear your voice twice. There was a resound from your side. You have logged in from one device, right?
Yes.
Yes.
You said
Yes. You said.
Okay, so I'm
I
sharing
logged
the
in
slides now.
once. Why this noise? Can you share my
Can you
slide
share
now?
my slide now? My slide now. My slide now.
Yes, give me a moment. I'm going to share your slides.
Hello? Hello?
Hello, Dr. Jin. You can present your talk now and let me know when to change the slides.
Okay, thank you. Hello, everyone. I'm Dr. Mrs. Jane Ijoma Ogochukwu,
Hello everyone, I'm Dr. Mrs. Jen Ijoma Ogochukwu.
here
Hello
to
everyone,
present.
I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen Ijoma Ogochukwu. Here to present. I'm Dr. Mrs. Jen
I'm from Pharmaceutical Microbiology and Biotechnology, Enugu State University of Science and Technology, Nigeria. Hello, can you hear me?
Hello, can you hear me? Hello, can you hear me? Hello, can you hear me? Hello, can you
Yes,
hear me? Hello,
we
can you
can
hear me?
hear your voice. Please start your presentation.
Should I go ahead?
Should I go ahead? Should I go ahead?
Yes,
Okay.
Okay.
Sorry, I'm Dr.
Sorry, I'm
Mrs.
Dr.
Jane
Mrs.
Ijoma
Jane
Ogochukwu,
Ijoma
Pharmaceutical
Pharmaceutical
Microbiology and
Microbiology and
Biotechnology, Enugu State
Biotechnology,
University of Science and Technology,
Technology,
Nigeria.
Nigeria.
I'm here to
I'm here
present
to
on
present on
formulation,
formulation,
characterization,
characterization,
and
and
evaluation of
evaluation of
anti-malaria activity
anti-malaria activity
of
of
artemether and
artemether and
doxycycline entrapped
doxycycline
in solid
trapped in
lipid
solid lipid
nanoparticles.
nanoparticles.
Second slide.
Second slide.
Here is my
Here is
presentation
my
outline.
presentation outline.
Third slide.
Third slide.
Introduction.
Introduction.
Malaria is a
Malaria
leading
is a
cause of
leading
death
cause
and
of death
illness
and
in
illness in
many countries,
many
particularly in
countries, particularly
Sub-Saharan
in
Africa,
Sub-Saharan Africa,
Central and South
Central and
America,
South America,
Southeast Asia.
Southeast Asia.
Here is my
Here is my
presentation outline.
presentation outline.
Introduction.
Introduction.
Malaria is a leading
Malaria is
cause
a
of death
leading
and
cause of
illness
death and
in many
illness
countries,
in many
particularly
countries, particularly
in Sub-Saharan
in
Africa,
Sub-Saharan
Central
Africa,
America,
Central
Southeast
America,
Asia, and
Southeast
Eastern Europe.
Asia.
And there has been an
And
ambitious
there has been
goal
an
to
ambitious
achieve
goal to
90%
achieve
reduction in
90%
mortality and
reduction in
morbidity of
mortality and
malaria,
morbidity of malaria,
particularly the one caused by
particularly the one caused
p-falciparum,
by
according to WHO 2009. Fourth slide. Here is a map
Here is a map
of the world.
of the world.
About half of the world
About half of
population
the
live
world
in area
population
at
live
risk
in
of
area
malaria
at risk of
transmission.
malaria transmission.
The red
The
region
red
here shows
region here shows
area where malaria is
area where
endemic.
malaria is endemic.
Fifth slide.
Fifth slide.
Overview of Artemether. Artemether is a herbal medicinal plant derived from Artemisia annua. It's an extract of artemisinin. It has an active metabolite dihydroartemisinin, which has a short half-life of 2 to 4 hours and bioavailability of 43.2%. It's used as an anti-tumor property. It has anti-tumor property and anti-malaria property. Doxycycline. Doxycycline is a semi-synthetic compound derived from oxytetracycline. It is highly lipophilic. It's highly lipophilic, which allows it to easily enter the cell with 18 hours, according to Okada and colleagues 2022. Hello? Can you hear me? Hello, can you hear me? Doxycycline is used, doxycycline has antibacterial and anti-malaria property. Sixth slide. Six slides. Six slides.
Hello, Dr. Jane. We can see the logins from your side, two logins from your side. So please log out from one device because there is a resound from your side.
Log out.
Okay, please continue.
Okay, thank you. Six slides.
Yes, please continue.
Okay, the next slide, please.
Okay, I'm changing it. A moment.
Why nanoformulation of artemether doxycycline? The use of artemether and lumefantrine in treatment of malaria has been associated with poor aqueous solubility, a rapid clearance of the blood circulation, systemic side effects, particularly in pregnant population, reinfection, and resistance. Given to this fact, a new design of therapeutic approach is of a great interest, and such new design is the nanoformulation of artemether doxycycline to achieve an optimum therapeutic efficacy with a little or no side effects. Next slide. Aim. Our aim is to develop, to characterize, and evaluate nanoformulation of artemisinin-based combination of artemether doxycycline that will have a long half-life and serve as a more effective and readily available alternative drug in the case of future resistance to current therapy or to current treatment, as the case may be. Next slide. Objectives. What are our objective? One is to formulate artemether doxycycline solid lipid nanoparticles. Secondly, we characterize these artemether doxycycline solid lipid nanoparticles. We looked at the in vitro release studies of this formulation, and also we looked at the in vitro and the in vivo antimalaria activity of the formulation. Next slide. Materials and methods. Here, we used shear homogenization technique in formulation of solid lipid nanoparticles, where the two lipids were used we use the phospholipid 90G and the purified goat fat. Here we used it at the ratio of 1:1 and 2:1. After which we melted it in a high temperature above the lipid temperature, and twin 80 was added, which we used as surfactants. About 1% and 2% to 80 were used. Also, we introduced the sorbic acid, which we used as a preservative, and sorbitol, which we used as cryoprotectant. They were also added in the formulation. The artemether doxycycline, which are our pure drugs, were also used at the ratio of 1:2, 1:6, and 1:10. After which, please, can we go to the next slide? After which the sample was lyophilized in a freeze dryer to get a water-free solid lipid nanoparticles, which we subjected to characterization. We looked at the FTIR, where we looked at the functional group of the formulation. We also looked at the encapsulation efficiency and the loading capacity, and we also looked at the particle size and the PDI of the solid lipid nanoparticles. In phase 2, we looked at the in vitro dissolution studies, where we looked at the percentage release and also the kinetics of the formulation. At the phase 3, we looked at the in vivo studies, where we checked the uh, percentage parastemia, the mean percentage parastemia, and also we looked at the phase 4, we looked at the in vitro molecular studies, where we looked at the cytotoxicity and the parastemia reduction, percentage parastemia reduction. In phase 5, we considered the data analysis, where we used various analytical tools to evaluate the study. The next slide. Here is the solid lipid nanoformulation composition. Here you can see the barges, the lipid ratios, the weight of the lipid, the drug ratios, the surfactant, the twin 80 we used at different percentage, the preservative, the sorbitol, and the percentage of the sorbitol and the cryoprotectant used. Next slide. Here is our results. Here we after the formulation, we went straight to check the percentage yield. Here in percentage yield, we observed that we had an increase, or an increase in percentage yield of the various formulation. You can see that in batch 1A and 1B, we had 83.7 and 77.8%. And also we had a minimum percentage yield of 37.9 and 42.5%. This indicates that the percentage yield is inversely proportional, or let's say the percentage yield is high in the phospholipid content. What do we mean? We meant that the amount of the higher the phospholipid, the sorry, since 50% of phospholipid of the percentage yield is 83.7%, while at 66.6% of phospholipid, the percentage yield was 77.8%. Thus, we suggest that an increase in ratio of goat fat may result in greater yield. We also observe that the minimum amount of percentage yield in two badges which we used was as a result of the weight of the lipid against the concentration of the drug. However, there was no 100% yield, indicating that there may be a loss. There may be a loss as a result of reduction in the percentage yield, as a result of reduction or the wastage during production. Next slide. Here we observed the entrapment efficiency. We had high entrapment efficiency in solid lipid nanoparticle, both in doxycycline and artemether loaded SLN. We had both high entrapment efficiency, and also we observed that the entrapment efficiency in doxycycline was higher. Sorry, this slide is off. Solid lipid nanoparticle was higher. Okay. Hello? No? Back? Go back a bit. Go back a bit. Yeah, here. No? No? The next slide. The next slide. Entrapment efficiency and loading capacity. Okay, here the loading, we observed that we had both a high percentage entrapment efficiency in both doxycycline and artemether solid lipid nanoparticles. In doxycycline, we had 96 to 63%, while that in artemether we had 83 to 31%. And also, but that of doxycycline was higher than the artemether. The reason there was a suggestion that it could be as a result of the ability of doxycycline to complex with the lipid matrix and also precipitate from out of the nanostructure. However, the reverse was the case in the loading capacity of the solid lipid nanoparticles. We discovered that both the doxycycline and the artemether were both lower from 10.22 to 0.10 percent. However, the finding was consistent with Abo et al. 2016, which shows that the artemether and lumenfantrine loaded SLM had low capacities. According to a colleague, 2011, it may be due to the high concentration of lipid matrix used in formulation against the smaller concentration of the drug. Next slide. Here is the physicochemical properties of pure artemether doxycycline, phospholipid 90G, good fat and solid lipid nanoparticles. Here we can see the melting points, the color, the texture, the solubility, and the Landamers of the pure artemether doxycycline and the phospholipid used. Next slide. Next slide. Here is the pH of the, sorry, no, back. Go back a bit. Go back a bit. Can you please go back a bit? Okay. Here is the FTIR spectrum band of both artemether doxycycline, goat fat, and phospholipid 90G. And the solid lipid nanoparticles. Here we observed that the FTIR spectra that were found in both Artemether, the cycling growth fat phospholipon, was also present in the solid lipid nanoparticles. Here is the physicochemical properties of pure artemether, doxycycline, phospholipid 90G, good fat, and solid lipid nanoparticles. Here we had the samples. You can see that the melting point of the samples, the color, the texture, the solubility in 2% methanol water, and the land mass, which is the wavelength. Next slide. Here is the pH of the pure sample of artemether and doxycycline, pH of the pure sample of artemether, doxycycline, and the pH stability of the solid lipid batches when stored in a room temperature and a different time interval. Here we observed that the artemether was in less acidic medium, which is 5.97, and the doxycycline was in more acidic region, which was 2.22. And also here in the pH stability of the solid lipid nanoparticles, the batches of the solid lipid nanoparticles at stored room temperature and a different time interval, we observed that the batches in day one, we noticed the batches in that it was more in more acidic region. At second day, we noticed the batches were also more in the acidic region, but at more 1 month, we noticed a shift in the acidity, towing towards a less acidic medium. And we suggested that we attributed the slight degradation due to oxidation of the fatty acid of the lipid matrix on the exposure of atmospheric oxygen. And also we discovered that the drug loaded SLN have different stability compared to the unloaded SLN. The difference in stability was attributed to the property of the drugs. Generally, we noticed a stability in colloidal pH of the solid lipid nanoparticles, which could be as a result of the solvents used, Swin 80 used. Next slide. Here is the FTIR spectra of the main bands region of the artemether, doxycycline, goat fat, phospholipid 90G, and the solid lipid nanoparticles. Here we discovered that the major peaks observed in the FTIR spectra of the pure drugs, the lipids, were also present in the solid lipid nanoparticles. As we can see here, look at it, we can see here, both the bands were also found in the solid lipid nanoparticles, and although with modifications in the frequencies. Also, there was no outstanding peak in the solid lipid nanoparticles. As we can see, there was no outstanding peak here. When compared with others, there was no outstanding peak, which may be due to the lack of strong interaction between the drugs and the lipids used. In addition, we observed a low intensity of absorption of FTIR, and we attributed it to be as a result of the low drug concentration used. Next slide. Next slide. Here is the particle size, the polydispersity index, analysis of the formulated drugs. No. Go back a bit. Okay. Here is the particle sizes and the polydispersity indexes, analysis of the formulated drugs. This, according to a Emerger et al. 2012, the burst nano size is within the range of 100 to 1000 nanometer, while the PDI less than 0.1 is considered to be monodispersed. The PDI more higher than 0.1 indicates polydispersed with broad particle size distribution. As we can see here, the PDI here, the zeta sizer here is within the range of it is within the nano range. As you can see here, you see 344.3, 526.4, and the rest of them. And the high PDI was considered that PDI 0 higher than more than 0.1 was considered to be polydispersed. As you can see here, most of the PDI were more than 0.1, and the PDI 1.0 is for a highly polydispersed sample with multiple particle size, and you can see some of them were more highly polydispersed samples, as we can also observe here in the PDI. Next slide. Here is the in vitro dissolution study of the solid lipid nanoparticles. This shows the percentage release of both doxycycline and the artemether. Both figures show a high percentage of drug release in SGI of both doxycycline and artemether. But we observed a maximum percentage of artemether release, which is between 76.2 to 100 percentage at 24 hours, both in artemether and that of the doxycycline. And there was also also noticed that however, we noticed a maximum percentage of doxycycline release from 71.9 to 100% at 24 hours. According to Manadol et al. 2015, artemether is the most potent of anti-malaria drugs due to its high peak concentration between 3 to 4 hours, rapid elimination of immediate release between 2 to 4 hours, and also we noticed a high release of a burst release of artemether within 0.5 hours, as you can see here, which agrees which agrees with the potence of artemether in anti-malaria as an anti-malaria drug. Also, we also noticed that there was a consistency, as you can see, there was a consistent release both in artemether and in doxycycline. As we can see here, they were consistent release. This shows a consistent drug release, which was observed both in artemether and doxycycline, could be attributed could be beneficial for maintaining steady therapeutic level of the drug over an extended period of time. Next slide. Here is the in vitro release kinetics of artemether in the SLNs. Look at the in vitro release in vitro release kinetics of artemether in the solid lipid nanoparticles. Here, both in vitro release of both the artemether and the doxycycline, we investigated the kinetics drug release using 4 models. We used the zero order, the first order, the Higuchi, and the Cosmea-Peppers model. Here, from the result, the zero order, as we can see here, both in artemether and doxycycline, which is the next slide, it did not meet the criteria. The negative sign, the negative value shows that there is no relationship between the variables, the time and the concentration. Thus, the zero order was not best fit for the data, indicating a concentration independent diffusion control release. But there was a fair correlation in the first order batch of doxycycline and artemether. This is artemether and that's doxycycline, the next slide. There was a fair correlation. Thus, suggesting that a constant proportion of drug was released per unit time. Also here, the best fit was seen in Cosmeya-Pepper's model, where we suggest that the release was a non-fecal diffusion release. Non-fecal diffusion release. The in vitro data was the in vitro data obtained in the study were best fitted in Cosmeya-Pepper's model for all data sets, which was very high, indicating a good correlation within the experimental data. This confirmed that the drug release was true non-fecal diffusion controlled. This prolonged release helped in maintaining steady blood level of the drug, while the burst release that we saw previously in the release study could be achieved in could help to achieve faster therapeutic blood level. Next slide. Next slide. As the in vitro study. Here, the in vitro study, we looked at the cytotoxicity study. We looked at the cytotoxicity, the viability of the red blood cell, the viability of the red blood cell to our drugs. Here, we looked at the red blood cell. The viability of the red blood cell was ascertained by screening the cytotoxicity effects, as we can see here, both in the solid lipid nanoparticles and the pure drugs. We use the NG. The NG here signifies the negative control. The result indicates that the survival of the red blood cell exceeds 75%, as we can see in the charts here, for all the badges of the SLN and the pure drug, ranging from 77 to 100%. However, due to the low toxicity level, the cytotoxicity concentration could not be determined for any of the badge of artemether and doxycycline, thus suggesting that all the badges were all good potential source of alternative anti-malaria drug. All the badges we have were very good potential for anti-malaria drug. Next slide. At 5 microgram per mil. Next slide, please. Here is the mean percentage parastemia count. Here we consider the mean parastemia count. From the result, there was a decrease in the level of mean percentage parastemia in all batches of artemether doxycycline loaded SLN compared to the pure drugs and the negative control, as we can see here, compared to the pure drugs and the negative controls. Here are the pure drugs, and here is the negative control. Here also, the mean percentage reduction in parastemia count was high, range of 9.1 to 11.9% for artemether doxycycline loaded SLN, while the mean percentage reduction dropped to 88.5% for artemether. You can see that of artemether, and that of doxycycline, 8.0 of that of the, and 6.1% for artemether lumenfantrine, and that's 4.3% for that of SLN. The clearance of parastemia by the formulated artemether doxycycline loaded SLN proves the ability of the nanoformulation to permeate the cell materials of the schizones of the P falciparum, leading to a sustained clearance across all batches as obtained in the release studies above. Next slide. Next slide, please. Here is the in vitro antiplasmodial activity assay using cybergreen. Here, cybergreen was used to ascertain the IC50 of both the pure drugs, the pure drugs of artemether, and the batches of SLN. Here we notice that in the 50% inhibition of plasmodium using 5 microgram of using a concentration of artemether, we observed that the IC50 was 6.286 of that of artemether. Please, next slide. That of the SLN, the badges of the SLN. Please, next slide. The next slide. The badges of SLN, we observed here that the IC50 was 1.218 microgram per mil, and that is the next badge, which is the 6B. We observed that the IC50 was 5.069. Since IC50 is the half concentration needed to eliminate the plasmodium, and the lower the IC50, the better the drugs. The IC50 of the badges were lower than that of the artemether, pure artemether used. Here, according to Ngameya and colleagues 2010, antiplasmodium activity was defined based on the IC50 value. And as we compared the work done by Bentum et al. 2019, where a plant extract was used against the chloroquine sensitive strain that was considered to have the strongest antiplasmodium activity with the IC50 value of 9.5 microgram per mil. These badges were deemed to be a very good to have a very good antimicrobial antiplasmodial activity, and showing that our drug, that is the badges of our drug, were very good, had very good antiplasmodial activity. Next slide. Next slide, please. Next slide, please. Next slide. Next slide, please. In conclusion, the research developed artemether doxycycline loaded SLN. The formulation drug had the highest. Please move to the conclusion, please. The conclusion, please. Conclusion. In conclusion, we developed a solid lipid nanoparticles of artemether doxycycline, which serve as an alternative drug to conventional artemether lumefantrine. We demonstrated the benefit of using solid lipid nanoparticles or nanoformulation as an effective drug delivery system to enhance the bioavailability of pure soluble or hydrophobic drugs. We formulated a new drug alternative for individuals with fatty food intolerance, and we developed a drug that can be used both in treatment and in prophylaxis. Here are my references. Thank you so much. Here's the suggestion for further studies. Next slide. These are my references. Next slide. Thank you so much. I acknowledge the TED Fund for their support. I acknowledge those that helped me in so many areas, the Noguchi Institute in Ghana, and that of Brazil, the University of Brazil, that really helped in this work. I really say thank you to them all. Thank you so much for listening.
Thank you very much, Dr. James. And so by this we came to end of our webinar, and I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed it, and thank you. Have a nice day, and we'll be sending you the certificates along with the book at your email. Kindly do check and acknowledge the me.
Mm.